Good morning, everyone. My name is Susan Marquesi, and I'm the new Assistant Director for the Biological Sciences Directorate here at the NSF. I'd like to call the meeting to order, so welcome. We're pleased to see you all at this hybrid meeting, and I'd like to start the meeting by calling on Karen Cohn to remind us of the FACA rules that will govern this meeting. Karen?
Thanks, Susan. I want to remind everybody that this is a public meeting. The meeting is being recorded and is available live on Ustream, on YouTube. By participating in the meeting, you are consenting to be recorded. We remind you that messages in the chat are also recorded, so please use the chat only for essential purposes. The Assistant Director, Dr. Susan Marquesi, is the designated federal officer for the Bio Advisory Committee. Please do not disclose Privacy Act protected information or any non-public information that you may receive or have access to as part of your ACE member status. The Bio AC has no oversight or approval authority and cannot participate by statute or NSF policy in budgets, hiring or management of NSF personnel, pre-decisional matters pertaining to awards or internal policies, or management of proposals, or management of bio within NSF. The Bio Assistant Director can seek the advice from the Advisory Committee about many topics, including some of the ones that we'll talk today, in areas of scientific interest and trends in research, for strategic planning and support of research, education, or infrastructure, for strategic human resource development in education and inclusion, development of metrics, or strategic communication with external communities, to name just a few examples. We're looking forward to today's meeting, and I'll hand the meeting over now to Dr. Michael Ibba, who's chair of the Bio Advisory Committee.
Thank you, Karen. So welcome. First, some business. This meeting is being held as a hybrid meeting. Some of us are in the room here at NSF and other participants are on Zoom. For those in the room, we ask that you log on to Zoom on your computer or iPad, enable your video, but mute your computer audio. Enabling your video will allow your face to be projected in speaker view if you are presenting or in gallery view when we are in discussion. When you wish to speak, please activate the microphone in front of your seat. The red light will come on. This will project sound through the room system. When done speaking, and activate the microphone. For those on Zoom, we remind you to mute your microphones when not speaking. If you keep your video on, this will allow your face to be projected in the gallery view during discussions. To request to speak, participants should use the raised hand feature. I'm now going to call on each person from the roster, the participant group, to introduce themselves. I'll start with myself. My name is Michael Ibba. I'm a professor of biochemistry and the dean of the College of Science at Chapman University in California. And my research focuses on microbial stress responses and evolution. I will now pass the microphone over to Barbara Beltz.
Barbara Beltz. I am now actually emerita at Wellesley College, where I was the chair of the neuroscience department. I am now, my primary work is with the Sherman Fairchild Foundation, running their undergraduate science programs.
Thank you. Hank Bart.
Hello, Hank Bart, professor of ecology and evolutionary biology at Tulane University. I also direct a Biodiversity Research Institute at Tulane.
Tom Daniel, who was going to be in the room, but I'll let him introduce himself anyway.
Thanks, Michael. It's a pleasure not to be there. I am Tom Daniel. I am emeritus professor at the University of Washington in biology, and I currently hold a position as the CEO of a nonprofit science philanthropy, the Washington Research Foundation.
Scott Santos.
Hi, Scott Santos from the State University of New York at Buffalo. I'm a molecular biologist, evolutionary ecologist.
Kennedy Wakesa.
Kennedy Wakesa. I serve as Associate Provost for Alabama State University, and I'm also a professor of biology.
Is Suzanne Barbour on? I can't see her image. Okay, Suzanne can introduce herself later. Eric Grotewald.
I'm Eric Grotewald. I'm a professor in the Department of Biochemistry and Molecular Biology at Michigan State University. And we work mostly on plant gene regulation and metabolism.
Rob McClung.
Hello, I'm Rob McClung. I'm a professor and chair of biological sciences at Dartmouth, and I'm a plant molecular geneticist, and my studies focus on circadian rhythms in plants.
Gretchen North. You're muted, Gretchen.
It says, okay, how are you hearing now? Yeah?
Yes.
Okay, I am a professor of biology at Occidental College in Los Angeles, and I study plant physiological ecology primarily in arid regions.
Maria Pellegrini.
Good morning, Maria Pellegrini. Currently Executive Director of Programs at the W.M. Keck Foundation and formerly a molecular biologist at the University of Southern California.
Maria Uriarte.
I am Maria Uriarte. I am a professor and department chair in the Department of Ecology, Evolution, and Environmental Biology at Columbia University, New York City. I am a tropical forest ecologist, and I study the process of forest recovery from natural disturbance primarily.
Thank you. We're now going to go through the NSF staff. Simon Malcolm will be joining us later. Now we'll go through some of the division leadership. So from the Division of Biological Infrastructure, Jim Deschler.
Morning, everybody. Thanks for, it's good to see y'all. Jim Deschler, I'm the acting division director in the Division of Biological Infrastructure.
Katharina Dittmar.
Morning, everyone. Katharina Dittmar. I'm the acting deputy division director in the Division of Biological Infrastructure.
So from the Division of Environmental Biology, Alan Moore.
Good morning, everyone. I'm Alan Moore, Division Director, Division of Environmental Biology. I'm a rotator from the University of Georgia and evolutionary biologist.
Bernice Smith.
Bernice is sick today.
Okay. From the Division of Integrative and Organismal Systems, Denise Deering.
Good morning, everyone. Nice to see you. I'm Denise Deering. I'm a rotator. I'm a division director of Integrative Organismal Systems, and I'm a rotator from the University of Utah. I'm a physiological ecologist.
Charlie Cunningham.
Hi, good morning. Charlie Cunningham. I'm the acting deputy division director in the Division of Integrative Organismal Systems, also program director in Molecular and Cellular Biosciences.
Thank you, Maria. I noticed that Paul Turner wasn't added to the roster. I apologize, Paul. Would you like to introduce yourself?
I was looking forward to the day off.
Oh, well. No chance, Paul.
All right. Hi, everyone. Paul Turner. I'm professor of ecology and evolutionary biology at Yale University, and I direct our Quantitative Biology Institute and our Center for Phage Biology and Therapy. I'm a virus evolutionary geneticist primarily.
All right. Thanks, and sorry about that, Paul.
I apologize. It's my fault, Paul. I made the roster.
No problem.
Karen's taking one for the team a lot today anyway, but don't have to, Karen. We'll do it. So we'll come back to division leadership. For the Division of Molecular and Cellular Biosciences, Teresa Good.
Hi, I'm Teresa Good. I'm the division director in the Division of Molecular and Cellular Biosciences.
Bianca.
Good morning, everyone. I'm Bianca Garner. I'm the acting deputy division director, and my day job is a program director in systems and synthetic biology.
And then we have some bio-OAD staff, Brent Miller.
Good morning, everybody. Brent Miller, science advisor, Office of the Assistant Director.
Karen, would you like to reintroduce yourself?
Sure, Karen Cohn, science advisor in Susan's office.
Rada Gonzalez. Is Rada on?
Oh, she's with Simon. She'll get it.
Okay. Is Kim Albuquerque here? Yes.
Good
Good morning, everyone. I'm Kim Albuquerque. I am the Acting Directorate Administrative Coordinator.
Is there anyone who hasn't been introduced?
I think me. I'm Gail McLean. I am from the Department of Energy.
Gail, yes.
Yes, I got here a little late. I was running from another meeting. I'm the Acting Division Director for Chemical Sciences, Geosciences, and Biosciences, and I am also the team lead for Chemistry and Biochemistry in BES, and my background is plant biology.
Good morning, everybody. My name is Megan Lewis. I am the Acting Communications Specialist for Bio.
Okay, I think we got everybody. All right, so.
Susan didn't have a link, so she.
Okay, all right. All right, so thanks everyone. So we're going to deal with 2 items of old business. First, I have some information to share. You may recall that in a recent meeting, we heard a report out on the decadal review of the long-term ecological research program authored by the 40-year review committee. At that point, the AC voted to accept the report for publication on the BioAC website. Upon publication, however, a typographical error was noted that underreported the number of publications from LTER activity. That error has now been corrected, approved by me on behalf of the AC, and the corrected version of the report is available on the BioAC website. So no further action is required on that. The next item is we require approval of the May 2023 meeting minutes. So the second item is to approve minutes from our May 2023 meeting. I hope everyone has had the opportunity to review the minutes. First, I'd like to ask if there are any comments or corrections. If not, I request a motion to approve the May 2023 BioAC meeting minutes.
So moved.
Second, would anybody like to second the motion?
Second.
Any discussion? All in favor, raise hands. Any opposed? Motion passes and the minutes are approved. So now I'd like to turn over to Susan Marquesi, the new Assistant Director for the Directorate of Biological Sciences, for her bio update presentation. Susan.
Thanks, Mike. I'll just wait a second. Meanwhile, good morning, everyone. Thanks for joining us today at our September 2023 Bio Advisory Committee. It's really great to have you all here. Special thanks to those of you that have made it in person. It makes it much more enjoyable. And to those of you on Zoom, thank you for giving us your time. To keep us on time today, I'm going to be fairly short. I'm going to jump into my update on bioactivities since the last time we met. And then really we can devote most of our time to the agenda of today. So if I could have the first slide, which is an overview of what I'd like to do. Thank you. I'm going to give you a short presentation. As I said, I'm first going to cover some staff updates. You've already heard quite a little bit about that as people did introductions. Then I'll do legislative and executive branch updates. I'm going to give you some news about programs, but really just to highlight and introduce those that are relevant to today's agenda. So if there are other things you want to bring up, just let me know. And then we'll conclude with just a few fun highlights of science. Next slide. Okay, so staff updates. Let's start with the obvious here. I'm new. So I'm the new AD here, and I've been here just about 2 months. So this is my first time at the AC meeting on this side of the table. I was a member of the AC committee, of the advisory committee from 2014 to 2017, and did overlap with some of you who I guess are doing multiple tours of duty. So for those of you who don't know me, I'm joining NSF as a rotator from the University of California at Berkeley, where I'm a professor in molecular and cellular biology, MCB, and chemistry. So I've been at Berkeley my entire scientific career ever since I was a postdoc. And my research focus and training are in molecular biophysics and biochemistry. My particular interest and expertise is in what I would refer to as protein dynamics. I study protein folding, design, energetics, so I'm very much on the biophysics side of things. My field is a mixture of experimental and theoretical. I am an experimentalist. I use tools that span the gamut all the way from the development of single molecule experiments to study mechanical, using the optical trap behavior of macromolecules, to basic ensemble biophysics and biochemistry, all the way up to some very simple genetics. In addition to my training as a PhD, I also have an MD. I was trained in the NIH Medical Scientist Training Program, or MSTP, although I went straight into research and never went further in doing a residency or anything like that. As I told you, I previously served on the BioAC and have served on several COVs for MCB. But other than that, I'm really new to the NSF and the NSF ecosystem. I'm not yet fluent in NSF, but I am working on it, so I am compassionate about the difficulty with all of the abbreviations that we use here. So if I don't define them or somebody else doesn't, please raise your hand because it is frustrating, I know. And so I'm really excited today to hear from you as much as I can about what you think we are doing well, what we're not, what we should be doing. And your input is not limited to today. You all have my email and you can give it to me whenever you want. So I want to thank everyone who ran the ship in this interim, from Simon, who's going to come in a little bit and be right there, to Teresa, Karen, and Charlie for their service in lots of roles that they acted between Joanne's departure and my coming on board, which was almost 9 months. And from my perspective, they didn't just keep the ship afloat. They really steered it and drove it, and I'm very impressed. Everything is in very good shape. Thank you very much. So enough about me. You can ask questions later. Next slide. If I can just give you some other staff updates. You might remember Pat Serrano. She was the division director for DBI. She's, I understood, presented to you various times over the past few years. So she's a rotator. Her 4-year time came to an end, sadly for us. And a few weeks ago, she's now back at her home institution of Michigan State. And so I just want to formally thank Pat for her service and as DD for DBI. And it's apparent and obvious to me already, she had a tremendous impact on us, and we're thankful for that. Next slide is Jim, who is on Zoom today. Many of you know Jim Destrel. He introduced himself from his presentations as his day job is Deputy Division Director of DBI. But with Pat leaving, he stepped in as Acting Division Director for DBI. And while he's Acting Division Director, Katarina Dittmar has come over to act as as the Acting Deputy Division Director. And as she told you, her day job is a program officer in DEB, and she's acted before as Acting Triple D for DEB in the past. So thank you, Jim and Katarina, for that shuffle, and that seems to be going, it's been a few weeks, very well. Next. Okay, so my stepping on board has allowed a bit of a domino effect for people to go back to their day jobs. So Teresa went back to her day job as DD for MCB. And as you know, working with Teresa is Bianca Garner in her new role as Acting Deputy Division Director for MCB. You've probably met Bianca before because she's presented to this group on a few topics. And we are just now starting the search for a permanent Triple D in MCB, and we'll let you know hopefully next time what the results of that is. And then I want to thank and note Charlie Cunningham's new hat as Acting Triple D in iOS while Michelle is on leave. So thanks. So those are all the domino effects, and most of them are just shifting for a while. So let's move on to the budget. As you likely know, both the House and Senate have released their markups for the fiscal year 2024 appropriations. Both markups come in very close at about $9.5 billion for NSF, which is actually equal to the FY 2023 appropriations, minus some special supplemental funding we received from CHIPS and Science to fund new activities and to help stand up our new Directorate for Technology, Innovation, and Partnerships, or as we call it, TIP. So it's September. This is all very much moving parts, and it's the beginning of a process, and there's potential of changes to come, but this is as we understand it today. Okay, next. Okay, legislative and executive updates. Reports continue to come out in response to the EO, or Executive Order on Biotechnology and Biomanufacturing. I'm just going to highlight the two that are an integral part of our discussion that we're focusing on for the rest of this meeting. So NSF led development of a section of the report on bold goals for the U.S. biotechnology and biomanufacturing that focuses on cross-cutting advances needed to support R&D in areas including climate change and clean energy, food and agriculture, human health, and supply chain resilience. Teresa and Brent are going to cover this in much more detail later today, and they're going to include a lot of information or some information on our new initiative catalyzing across sectors to advance the bioeconomy, which we also call CASA Bio. So the other one up there on the slide is that NSF participated in developing the report on workforce development for the bioeconomy, and you're again going to hear more about this from Celeste Carter in the Directorate EDU on details of this report tomorrow. So you'll hear more about that. Not shown on this is that the NSF co-chaired the group that produced the report on vision, needs, and proposed action for the data for the bioeconomy initiative. That report, which is still in process, is not public yet, lays out the data and data infrastructure needs to support a robust bioeconomy and proposes actions to address those needs. I want to thank the members of the AC that provided input to the interagency working group that developed that report. So thank you. Next slide. Okay, so in preparation for our meeting of the next 2 days, I want to give you a quick overview of NSF investments in the bioeconomy, because that's what we'll be focusing a lot of our agenda on. So as I'm sure you know, the NSF invests across the continuum of basic, starting in blue on the left on this slide, research, to translational research, which is gold on the right. And these investments span our directorate as well as other directorates all the way over to the far right, where our Directorate for Technology, Innovation, and Partnerships focuses. These investments align with the bold goals that Teresa's going to cover later. So what I want to do is just highlight a few of these programs on the next few slides that might come up during our talk or that we might focus on. So next slide. Okay. In terms of education and training, there are a variety of programs that specifically target training opportunities in biotechnology. One of our major investments in this space is the Advanced Technological Education, ATE program, Innovate Bio. It's a center-scale activity funded by ATE, supports the development of community college programs in the biotechnology area across the country. Currently, there are 117 college programs that Innovate Bio serves, with 89 K-12 schools. The program develops and disseminates materials for community college instructors to help connect programs and students to industry employers. Biotechnology programs at two-year colleges are responsible, as I'm sure you know, for educating many of the technicians who work in the biotechnology and life sciences industries, highlighting the importance of this program. Additionally, there is the newly launched Experiential Learning for Emerging and Novel Technologies Excellent program, a joint effort of the Directorate for STEM Education, or EDU, and TIP, that seeks to support learning opportunities for individuals from diverse professional and educational backgrounds that will increase access to and interest in career pathways in emerging technology fields, including biotechnology. Next, we have the Research Traineeship NRT program that supports interdisciplinary evidence-based traineeships that advance ways for graduate students in research-based master's and doctoral degree programs to pursue a range of STEM careers. Over the past 5 years, the program has made 19 bioengineering and biotechnology-related NRT investments. There are also programs focused on enhancing research capabilities of minority-serving institutions and HBCUs, including NSF's Centers of Research Excellence in Science and Technology, CREST program, and HBCU Research Infrastructure for Science and Engineering, HBCU RISE. So that's just an overview of those. And now if we go to the next slide, So this is, just want to make sure I'm in the right place. So this is BIO's particular support and workforce development. It's a very dense slide. It's just there for you to look at, and you've heard about most of these things, and you can refer back to it as we go through the next 2 days. You've heard about BIO's training programs, including the postdoc program, our new research capacity building program called BRC Bio, and our postbac program, RAMP. So more information about each of these programs can be found in your materials, and you'll have an opportunity to reflect on these tomorrow in our session on bio workforce development. So this is really just highlighting this. We'll also talk about infrastructure later in the meeting, so I just want to give you a quick bit of info on our new BioFoundries program. If you can go to the next slide. BioFoundries is a new NSF-wide initiative that is designed to accelerate advances in the biological sciences, chemical biology, biotechnology, and bioengineering via access to modern infrastructure, technology, and capacity. The goal of this solicitation is to support the establishment of infrastructure that focus on the innovation pipeline and research, while ensuring broad access to the research resources, training, education, and outreach to catalyze our understanding of biological or bioengineered systems, and to support the design, build, test, learn pipeline for rapid translation of basic discoveries into applications. The research aimed to integrate advances in modern biology, chemical biology, biotechnology, and related fields with innovations in automation, high-end measurement infrastructure, nanotechnology, microfabrication, integrated testing and data acquisition, and AI-enabled analysis and automation of the scientific discovery process. It addresses needs outlined in several of the reports in the recent EO on biotechnology and builds on existing NSF investments and programs, for example, materials innovation platforms, EDGE, NeuroNext, RECODE, partnerships with the DOE, Agile BioFoundry, future manufacturing, and cyber physical systems. So Sri Raghavachari will provide more information in the research infrastructure session this afternoon. So that's just a brief introduction. Okay, next slide. So I just want to close with some fun recent science highlights for everyone. So first click, yeah. So funded by DBI and IOS, researchers at MBL, or the Marine Biological Lab, found that two-spot octopi adapt to seasonal temperature shifts by producing different neural proteins under warm versus cool conditions. So the octopi achieved this amazingly through RNA editing. And this rewiring likely protects their brains. So the work will now move on to determine if octopi and other cephalopods use RNA recoding to adapt to other environmental variables, such as perhaps low oxygen availability or varied social environments. Next. In research supported by the Evolution and Ecology of Infectious Disease program in DEB, conducted a pilot study to determine the effects of removing vegetation from water access points in West African villages. So this removal resulted in reduced rates of schistosomiasis, which is a disease caused by trypanosome. Additionally, the removed vegetation was composted, resulting in a fertilizer that increased onion and pepper yields and can be used as feed for sheep that is somewhere between 41 and 170 times cheaper than standard, depending on the amount of vegetation supplementing traditional feed. Next thing, supported by MCB, researchers at Wake Forest University have found that ethylene is an important factor early in seedling development that has long-lasting effects on plant growth and tolerance to stresses after they transition from darkness into light. When the seedlings treated with ethylene in the dark for several days were subsequently given light and then had the ethylene treatment discontinued, the seedlings did better than controls. This suggests that ethylene treatments could help improve yields across a range of crops without sacrificing stress tolerance. Finally, I just want to highlight across bio and how our divisions connect. This is research now co-funded by MCB, DEB, and DBI. And what they did was to show that a minimal cell, so you probably know about this, it's a cell with only the essential genes in its genome, maintains its ability to evolve and optimize its genome. So this ability brings us one step closer to using these cells in biomanufacturing approaches and other efforts. And it's a beautiful example in my mind of life finds a way and illustrates our ability and mission to use life to improve life. So with that quick introduction, I think I'll pause, and I'm happy to take questions and answer them as best I can, or turn to my colleagues who can probably give you more factual answers about what we do here, or give you my own opinion. Thank you.
All right, thank you very much, Susan. Before we go into the questions, Suzanne, would you like to briefly introduce yourself?
Hi, everybody. I'm Suzanne Barbour. I'm at Duke University, where I'm Dean of the Grad School, Vice Provost for Graduate Education, and Professor of Cell Biology, and just very excited to be part of this important group.
All right, thanks, Suzanne. Okay, questions for Susan? Come on, it's her first time. Don't go easy on her.
Suzanne has her hand up.
Suzanne?
Well, I'll break the ice. I'm also on CEOS, the Committee on Equal Opportunities in Science and Engineering. And we last year put together a report, a review of the EPSCoR program. And we're working right now on a report that focuses on rural STEM education. And one of the things that's come up in our conversations that might be relevant to biofoundries is the issue of particularly rural environments and the degree to which schools and community colleges and other entities have access to broadband internet, and whether one can truly ensure that folks in rural areas have the opportunity to aspire to careers in STEM, even if they're just careers in the skilled technical workforce, without having access to broadband internet. So I only say that to say that I think there's another layer in the biofoundries conversation that I hope will be considered.
Thanks for bringing up that important point. And I personally, I hadn't thought about that directly in that way. So it's good for us to, I don't know if anyone here has anything to add for that, except for I take your suggestion, not just for the biofoundries, but just to bring that up in general. I actually had lunch with the chair of your committee just last week, so learned a little bit more. Thank you.
Rob.
Much of what you were talking about has to do with workforce development. And as I'm sure you're aware, there seem to be changes in the self-perception of the workforce, notably graduate students and postdocs starting to unionize and so on and so forth, creating significant upward pressure on salaries, but the budget hasn't increased. So what are your thoughts on how we're going to go ahead with this?
Yeah, thank you for bringing up that important point. I think about it a lot. I don't have solutions yet. So there is the budget to us. I assume that's what you meant when you said doesn't increase, right? We have a certain amount of money, and it's not up to us to, how do I say? The salaries are usually governed by the individual institutions. And so I want to remind people that when they write their proposals, they should ask for what they need. That doesn't really address your question of the limited funding, but it is very difficult for lack of a better word, to supplement that funding if your grant is not covering the full required salary for a postdoc. So it's true, given the amount of money people have now, they just are not going to have the amount of personnel, which of course brings up a lot of issues about the what can be in one grant and what can be accomplished. And And I think we have a big role to play and think about this going forward, but we're just a small part of the whole ecosystem in this. And so I know the NSB is trying to think about how we do things. I'm planning to meet with the NIH, with the NIGMS particularly. They do have set salary scales, and they are moving them, but how we as a whole ecosystem and science, and what does it mean? I don't know if anyone else read, is it the most recent Nature that had the issue of, about the postdocs in UW? It was more of a story. There wasn't that much data in there, to be honest, but is what will the horizon look like, particularly for postdocs? And how many postdocs will there be going into the future? Things are changing. I just want to remind you that I come from the University of California system, so I've experienced this change firsthand over the last year. And I think that compounded with the pandemic have made being a graduate student a very different experience than we all went through when we were in graduate school, which also adds an added layer of how we mentor people who are not having the same experience we are. And they are demanding to be mentored in their training. So there's this sort of push and pull between being an employee and being a trainee. And so I don't have any answers. If you have suggestions, I'd like to hear, but I can promise you this is definitely on my agenda to think about going further.
Hank?
Thank you. Yeah, my question is about just more general costs, overruns in projects. I notice about my own projects, but I suspect a lot of people are experiencing this. As we were coming out of the pandemic, and there were supply chain issues and inflation, those of us that had grants that were where the funding was fixed, experienced shortfalls. And I don't remember reading how NSF has been advising people to deal with that kind of situation. Can you just comment on that?
I cannot, but Teresa, can you?
So I can start. I think that whether it is salaries or it is like that, there were, during the middle of the pandemic, we had authority to provide supplements above and beyond the 20% without review. That authority has gone. But, and so there's always the opportunity to interact with your individual program director to request a supplement, but we don't normally provide them explicitly for cost overruns. That's not the policy. And so that becomes a little challenging. Plus, our budgets are fixed, and everyone's cost of, if we are to fund the real cost of research, which is a core value, our costs are fixed. And so what we have in general been advising people to do is to submit their renewal early, because that's really the best way for us to get you the next set of funding you need. But there are, on a local level, there's lots of individual variations on how we do that. I don't know if any of the rest of you wanted to contribute to that.
So maybe I can ask, oh, sorry, Paul.
Since Mike asks us to deliver difficult questions, here's one. So our trainees demand jobs outside of academia often. Susan, what are your thoughts on the relationship between academia and, say, industry? So can you consider some of the pros and cons of this? Have you grappled with this much in terms of what can we do safely and prudently with partnering with industry? Because this is the goal for more and more of our trainees to work in biotechnology. What do you think?
Yeah, I mean, and yes, and I think about this a lot. But I'm not quite sure I know what you mean by partnering. There's partnering in being able to make sure our students are trained for the careers they want and have that experience, which is outside of most of their mentors' experience. There's that issue. Are you talking about a financial partnership along the way? That's what I was confused about your.
Yeah, it's the limited budgets of the
right.
NSF awards. And is there any way to capture safely through partnerships, because this is also verging into conflict of interest.
Exactly. Yes.
That's the difficult part.
Yeah, no, and I, you think about that a lot. Sorry, Teresa, you want to add on before I step on my toes, or?
I don't want to.
No, go ahead.
So there's a couple of ways that we do partner with industry already. Some of it is through a program where we, that some parts of bio participate in the Industry University Cooperative Research Centers program, where NSF provides primarily the administrative support for those centers, but industry provides the rest of the money for those centers, and it's a great opportunity for student training. Another of those kinds of opportunities that we get that kind of training with industry are through some of the science and technology centers that we support. So if you talk to, say, Charlie or others around the table in terms of the science and technology centers that they manage, that they can talk to you about what kinds of opportunities there are there. There are things like intern supplements, as well as this experiential learning program that is new with TIP. I don't think we necessarily know the great details about that. And then I think that Brent is busy all of the time thinking about how to develop new partnerships. One of them he's trying to develop is with one of the Manufacturing USA institutes, where we would enable our researchers to partner with this institute that gets the bulk of their funding through kind of one-to-one cost share with industry. And so we're exploring more and more opportunities in that space.
I apologize, just a quick follow-up. I apologize for not knowing, but is there some special work going on with minority serving institutions, HBCUs, tribal colleges, especially in this regard, since so many of our students are trained in those realms and they want to go into well-paying jobs? Not that academia doesn't pay well, sometimes it does. But what do you think?
That would be a great opportunity, a great question for Celeste when she comes to visit us. I don't know the answer.
Thank you.
So I just want to add, Paul, that, and then, sorry, Jim, I'll call on you, or you'll call on, is that this trend may be new for biology, but this has been the case in engineering and chemistry. They never thought all of their graduate students were going to be clones of themselves, and they were going to provide the workforce in other ways. And so we have a lot that we can learn from our partners across the other directorates in doing this, to make sure that the students are prepared, aware, explore the careers properly. And it's also something that is on my agenda to try to move forward with. So thank you. Sorry, you.
Jim?
Yeah, just to add a little more, Paul. We think about it a lot internally, and there are administrative hurdles. TIP is new, and they're thinking about workforce development, and we're doing things like cross-fertilization, cross-details between TIP and bio in this space. And we're looking forward to sort of how to, for example, link our broadening participation track in the postdoc program to more opportunities in that space from an NSF spearheaded way and through partnerships, since TIP is partnerships as well, right? So we're sort of excited about some recent developments in that space, but nothing's really flushed out yet to talk about.
Thank you.
So I'll add a couple of points a couple of things to what Paul was saying. So in terms of these students wanting to go into different careers, I do think that, for example, when I ran a T32, the NIH, I think, are a little ahead on this. They require that you include quantitative bio training, whether or not the person has access to that through their lab. You're required to add that in. You're required to add scientific communication and writing skill sets. Some of the things that students struggle with when they come out of their PhDs, if they go into look for jobs in industry. And that's where I think that there can be support that's useful. In terms of helping with internships and other possibilities through some of the institutions that you mentioned. Now, when I'm at a smaller institution, I see that there's an infrastructure bottleneck in so much as this usually requires going through contracts, things to do with the relationships with the companies and so on. And some of the smaller schools struggle with this if the capacity is put upon them. So I think the meaningful workforce development needs to also include some of the rather boring stuff to be productive through this. I know it's not very exciting, Susan, but
Yeah, no, you're totally correct. There's another issue is, of course, the funding cycle for the companies that are taking the internships is on a quarterly basis. And so we're used to planning, and we work, meaning the research community, we work on a very slow time scale compared to them. And so to set all of those logistics up gets very difficult because they don't know whether they're going to. So those are things we have to work out. I agree.
Maria?
Yeah, I'd like to follow up on this thread of postdocs and how much they get paid and so on. We're seeing also where students moving into carbon startups, data science jobs, things that we weren't seeing before because primarily monetary reasons. I mean, they just get paid 3 times what they get paid as a postdoc. So it's a very difficult comparison. And then, of course, the impact. But what this means, and I've seen this in my own work and also discussed it with many colleagues, that this is leading to a kind of a shortage of postdocs, which has been very instrumental to the research enterprise. So what is NSF and more broadly, the federal government thinking about these postdocs? I was surprised. I mean, I just hired someone, and pretty much I would say 90% were postdocs from Europe or Asia, almost no American applicants, which is fine. I mean, I don't need to say, but in general, it's just getting harder to get a postdoc.
Yes, I think that's been documented everywhere. There's also, but what is new is, in my opinion here, is the postdocs quitting with 2 weeks notice because they got a job, right? And this is they realize that that's their future and that's their prerogative, but it does make it difficult to complete your goals in your research. So I think this is beyond our, but as a nation, we're going to have to redecide what postdocs' roles are, how many we should have, what the lab size should be. And of course, there's the compensation, which all of the universities and the unions are taking care of. So it does remind us that postdocs are mentored and that we have to also provide something besides just their workforce. So these are issues I personally think a lot about. But if you read this recent article that just came out in Nature, it also talks about what the are we going to just get by with less postdocs? I don't know the answers to these questions.
Suzanne?
So years ago, NIH tried to grapple with this in some ways. There was discussion, I want to say it's probably 10 years ago, maybe longer, about only supporting trainees on training grants, with the idea that NIH would expand its support for quote unquote professional laboratory staff. The idea being that there's a cohort of people who don't want to be PIs, but they do want to be in the lab. They do want to be close to the research. They want to attach themselves to a laboratory and stay with that laboratory for their entire careers. But there's really no space for them right now. And I know that conversation never really went anyplace at NIH. It was a conversation at one point, but maybe it's time to resurface. That's the concept of having a role for quote unquote professional staff. And I know when I talk to colleagues at some of the places where I've been, they tell me very often they have postdocs who love being in the lab. They love mentoring graduate students. They love mentoring undergrads. They love even helping to write grants and papers, but they don't want to be PIs. And right now there's no place for them. So just another, when we think about the workforce, that might be a piece of the workforce that we haven't really addressed that might be appropriate for us to think about now.
That's a good suggestion, and we are having a workforce talk. Just to let you know, I personally was on that reconsidering the bio workforce, whatever that committee that wrote that paper. I was on that, so we can dig it out. And that was at the beginning of IDPs and things like that. Yes.
Tom?
Just pulling on this thread some more. I have a growing concern about the dilation of graduate careers. So if we interject post-baccalaureate training and doctoral training exceeds 5, 6, sometimes more years, and you can look at the statistics nationally, they're growing in length. That's dissuading people from entering STEM. And I think there has to be some national discussion about why we have to have such long doctoral training programs relative to, say, many in the EU. And that also brings people earlier in their career into the postdoctorate, so they're not as desperate. So it all is part and parcel. So I'd like to throw that into the wrench. Okay.
Yeah, probably beyond our AC, but yes.
But it's particularly egregious in bio relative to some of the other science domains, like say relative to engineering.
So I'll add one to your point, Tom, about this dilation. I always thought it was interesting that NIH spend a good deal of money to keep people doing research after they get their MD if they want to. They have all those fellowships to try and keep you in the lab. If you from the MSTPs, once you get your PhD and your MD, and they do. Whereas we just like kind of let people drift and pray. It's going to work. Rob.
To continue pulling on that thread, Tom, in some ways, I think what we're looking at is a self-inflicted wound, as one experiences over and over again when you get the reviews back on a manuscript that requests another year of research. So if the graduate student needs to get a good paper in a reputable journal, we need to persuade the reviewers to be more reasonable, I think.
I think just like the workforce is undergoing a huge transition, I would say that the publication arena is also going through a huge transition. And what that word means, we don't have to discuss it here. A reputable journal is not, that's also in discussion right now. So we are in the midst of a big change.
So if there are no more hands, I will say that when we talked about this before the meeting, everything that we're trying to do in this meeting depends on having people to do this. So workforce is kind of top of the agenda. And I think this is where, I won't say for once, I think we have a lot to offer when we meet with the director in terms of all of our experiences and what we can say about trying to come up with something which we think can make a difference. I think that's kind of key at this point, because the current model basically is not sustainable. We can all see that. The wheels are coming off. So it's an opportunity, I think, to do some good to the point that Suzanne and Paul have raised about where this work is done. It's a pretty big issue that hasn't been as successful, I would say, as it could have been. It started, but it's never really moved the needle in the ways that we all would have hoped. So I think this is going to be a good meeting. So I just put that out there because I think that this is a good point, a good string to keep pulling on as we go and meet with different people so that we can try and develop something. Or is it me again? Okay, I think we have a break now because the next bit doesn't start for 20 minutes. Is that correct? We're going to take a 20-minute break and come back at 11:15.
Well, when I stop the recording, so you.
I have to use this.
Yeah.
Okay, if we can get back on track, I've got to wait for the YouTube. There's the YouTube. Okay. So in June of this year, the Division of Environmental Biology hosted a committee of visitors. Hank Bart was the AC representative on that committee and has agreed to provide a brief overview of the COV's findings. Hank, over to you.
Thank you, Mike. So, just to give you a little bit of information about the people that served on the committee. So the committee was very ably led by Ann Yoder, and you can see all of the other members of the committee here. The 2023 DEB COV was charged with considering performance of DEB in two primary areas. One is assessing the quality and integrity of operations, including technical and managerial matters pertaining to proposal review and recommendations. And the second was assessing whether the portfolio of awards was appropriately balanced. So within those two areas, there were a number of different questions, assessing the quality and effectiveness of the program's use of the merit review process, questions concerning the selection of reviewers, questions concerning the management of DEB, questions about the resulting portfolio of awards, other topics. And the period that the report covered, that the study covered, was 2019 to 2022. So in terms of questions regarding the quality of the merit review process, the COV evaluated 384 proposal jackets, which resulted in reviews of 215 unique grant proposals. The number's lower because a number of the proposals were collaborative, multi-institution, and multi-investigator. Proposals that we considered will receive between 3 and 9 reviews. The average was between 4 and 6. In general, the COV found the review process to be thorough and appropriate, with substantial thought given to the representation of both ad hoc and panel reviews. And I'm going to say more about that a little later on as well. One of the big emerging issues was COVID-19, how it forced NSF personnel and investigators to abruptly assume new protocols for proposal evaluation, general working conditions, and work-life balance. The COV was deeply and positively impressed by DEB's response to this global crisis, with virtually no diminishment in proposals evaluated and grants distributed, nor in the quality of the review process. One of the other topics we considered were both merit review criteria addressed in reviews, and was reviewer feedback substantive. Just a commentary first, the COV noticed considerable improvement in understanding of the broader impacts criterion among investigators, the amount of page space devoted to broader impacts and proposals, and critical review and appreciation of broader impacts plans by reviewers. In terms of reviewer feedback, reviews differed considerably from non-substantive and actually generally the least helpful kind of feedback to highly substantive, which was also generally the most helpful, even when that information was critical, that review was critical. So we saw that full spectrum of different degrees of substance in reviewer feedback. In terms of reviewer training, we learned from program officers that some DE programs offer training to panelists, but not to ad hoc reviewers, which is important for DEB because it involves lots of ad hoc reviews and review of its proposals. And then just as a final commentary, that the COV sends some potential for inequity for researchers at institutions with less sponsored projects infrastructure. And hopefully this is something that programs like Granted will help to alleviate. Another question was, does reviewer feedback provide rationale for the award decline decision? The committee of visitors observed instances where panel recommendations and the funding decision didn't align. However, the rationale was provided in a review analysis document that's included in the jackets, but wasn't always articulated clearly in the panel summary, hence the lack of alignment. So the recommendation the COV made was, to the extent that information in the review analysis can be shared with proposers via a process called PO comments. This can serve as a mechanism for alleviating concerns when proposals are declined, and it could ultimately improve investigators' information, provide investigators information that could increase their chances of success on resubmission of the proposal. Some of the additional comments on the quality and effectiveness of merit review process. There was lots of discussion in the COV about virtual, in-person, and hybrid panels. There was lots of discussion about the pros and cons of those different kinds of ways of getting people together. And a lot of this was stuff that came out in the pandemic and post-pandemic decisions about how the whole panel process was changing. Virtual panels had the advantages of greater panel accessibility and diversity. They were less costly. There was a lower carbon footprint in the sense that people weren't flying to participate in panels in person. Fewer proposals were handled per panel, but POs commented that reviews were more thoughtful. The Zoom breakouts tended to neutralize really strong personality effect in panel discussions, which was also seen as a positive. But some of the drawbacks was, naturally in a virtual panel, there's limited interactions among panelists and POs. There's limited time for panel caucusing, or limited opportunity, and therefore there was less consensus. And there was more and there was a possibility that panelists would be more distracted because they were looking at email and things like that. And then, as I mentioned earlier, there's less time overall that would be devoted to those virtual panels. In-person panels have benefits. According to the program officers, 2 to 3 times more proposals are handled. There's networking opportunities, less distraction, more engagement in panel discussions. But some of the drawbacks that we heard about was greater workload for staff. And then hybrid panels also would double the workload for staff compared to virtual in-person panels. In terms of selection of reviewers, first some commentary. The COV commended DEB for its success in including ad hoc reviewers. In addition to panelists for proposals in many of its programs, the exception was what we refer to as special programs that are generally just reviewed by panelists. In 2022, 62% of DEB proposals were reviewed by ad hoc experts, compared to 25% NSF-wide. That was information provided in a 2023 self-study. And actually, the acceptance rate of ad hoc reviewers was 58%. And, we gave the DEB a lot of credit for its effort actually soliciting reviews from ad hoc reviewers and the rate in which they responded favorably. A recommendation that was made was updated analysis of how the co-review process in some of these special program types of opportunities affects the selection of reviewers, both ad hoc and on panels. And it suggests more transparency in decision-making process for co-reviewed proposals. In terms of program management, the period reviewed by the 2023 COV covers the transition and no submission deadlines, no deadline submissions, for proposals within certain DEB programs. Communications with DEB staff and program officers were largely positive as it relates to the no deadlines impacts, though there was acknowledgement that this led to changes in work processes and distribution of effort over the course of the year. POs highlighted a lower number and generally higher quality of proposals received, and staff members noted reductions in administrative processing workload. One of the emerging issues, the lingering effects of the pandemic. The lingering effects of the pandemic were a major topic of discussion during the COV, and there was significant confusion and no small measure of anxiety evident among the POs and staff regarding the path forward in DEB operations. The introduction of hybrid and virtual and in-person panels, review panels, along with the loss of key elements of logistics support, the example being like the availability of catering for panels, has resulted in increased workloads for existing staff. In terms of the responsiveness of the program to emerging research and education opportunities, a specific example of DEB's response to emerging research opportunity is its involvement in funding the funding program Biodiversity on a Changing Planet, which is consistent with the whole emphasis on global climate change. DEB scientific competencies are crucial for increasing understanding of the impacts of global climate change on the biosphere and facilitating the transition of knowledge gained from adaptation and mitigation strategies in collaboration with NSF's directorates, such as the Technology for Innovation and Partnerships Directorate, TIP. There was recommendation that with the recent increase in special programs and special program funding, it's important that DEB review how monetary and time budgets are divided between core and special programs. There was a lot of conversation about this as well. There was concern that there was an uneven division of funding across those two different kinds of programs, but our analysis didn't really bear that up. In terms of responsiveness to the program, the previous COV comments and recommendations, again, the CV commended DEB for its thorough and effective responses to nearly all the recommendations in the 2019 COV report. The COV was particularly pleased to see that DEB successfully encouraged more serious consideration of the ad hoc review during panel deliberations, which again was something that was borne out in ad hoc reviewer participation and reviews for the current COV study. One other issue was that the proportion of proposals submitted by PIs at MSIs had shown a steady increase until 2000, when there was a steep drop, most likely related to, in proposals submitted, most likely related to the pandemic. However, according to the Department of Education, in 2020, 20% of higher education institutions qualified for MSI funding, while only about 13% of DEB submissions came from PIs at MSIs. And this suggests that more work is needed in this area. In terms of the question, are awards appropriate to the size and duration of the scope of projects funded? A commentary in the report was that DEB is to be commended for its continued support of the community through the size of the awards it makes. Only 5% of awarded PIs received less funding than requested in their proposals in our study period. And that's an exemplary track record and suggests that funds are appropriate for the size and scope of awarded projects. The average duration of projects has stayed relatively steady over the 4 COV study years, while award amounts have steadily increased, with the exception again of 2020. And the decrease is likely due to post-pandemic supply chain and inflation issues and increases in personnel costs. Another question was, does the program portfolio have an appropriate balance of awards to different types of institutions and early career investigators? 2 different questions, actually. For most of the institution types characterized 2-year and 4-year schools, non-traditional, master's, PhD, et cetera, DEB's 4-year funding rate was generally proportioned with the overall submission rate of PIs at these institutions. And again, DEB is to be commended in maintaining a well-balanced portfolio overall, in which a large variety of institutions appear to be proportionally represented awards. In terms of new PIs were funded at disproportionately lower rates than the overall DEB award rate over the years 2019 to 2021. And a recommendation was made for continued encouragement of emerging PIs to consult program offers throughout the development proposals and to discuss review process and any resulting reviews. The question that we considered was, does the program portfolio have appropriate participation of underrepresented groups? The answer is an emphatic yes. The number of minority-led proposals increased over the study years, with the notable exception of 2020, which saw a disproportionate decrease in both the number of minority-led proposals submitted and the number of minority-led proposals awarded compared to the overall pool of proposals. However, in the 2 study years following 2020, proposals submitted by minority PIs and co-PIs rebounded and even surpassed the number in 2019. And to the question, does the program portfolio include projects that integrate research and education? First, the commentary that a number of special funding programs incorporate educational components, including research and undergraduate institutions, career, REU, research assistantship for high school students, research for teachers, and ROA programs. So RUIs in particular retained a 50% award rate in 2020, but decreased to just 10% in 2021, perhaps as a consequence of COVID-19. But RUI award rate rebounded to 31.5% in 2022. And supplements remain the main source of funding for the educational program, educational components of most of the grants that we evaluated. So the recommendation was made that the COV increase training around supplemental funding opportunities in funded proposals, perhaps through workshops or DEB office hours. And PIs should be encouraged to increase the education component of all grants, not just those specialized in education. And I think this is the last one. Is the program relevant to national priorities, agency mission, relevant fields, and other constituent needs? Again, the commentary, NSF has done a commendable job responding to national priorities, preparing a skilled workforce to meet the challenge, and increasing representation of women and minorities in STEM. The goals have been achieved while still retaining a focus on core DEB programs and innovative core research. Good examples of DEB's central role in this cross-cutting multi-agency is its involvement in a cross-cutting multi-agency ecology and evolution of infectious diseases program, which funds research on infectious disease modeling, prediction, and forecasting, and also DEB's involvement in supporting state-of-the-art infrastructure, basic research, and open data products stemming from its investment in the National Earth Observatory Network. Additionally, DEB played a key role in responding to COVID-19's pandemic by funding a number of rapid and eager grants. So I hope that all came through clearly. That's all I have. And I'd be happy to take any questions.
All right. Thanks very much, Hank, for that very comprehensive report. Does anybody have any questions for Hank about the COV report for the DEB? Yes, Paul.
That was great, Hank. Thank you for the report. Thank you to the committee for their work. I think I was a representative from BioAC at one of these prior reviews. I think it was actually DEB, but I know how difficult it is. So I have a weird question that relates to ad hoc reviewers, which I think there are a lot of upsides to it, and you summarized some of it. And I apologize to the committee if I've asked this before, but I have this weird paranoia about the total number of reviews and whether there's a downside to it. Because of, if you ask 9 people for their opinion on something, you're likely to get somebody who hates it. And that's just a probabilities thing to me. So on the one hand, I have really enjoyed the ad hoc review process and the comments I've received from ad hoc reviewers in the past, but has anybody ever done kind of an assessment on the correlation between the total number of reviews you receive and whether there's some bias? I see some people chuckling about this. So either this has been looked at and dismissed, or there's a little hint. I don't know. Educate me on this. Has anybody looked at this? We talked about a downside to ad hocs.
I was chuckling, Paul, because it sounds like a way to avoid jury service, but I'll let Hank answer.
Maybe.
Yeah. Sorry, Brent.
So we've looked at a number of different measures, and that is one of them that we've not looked at. And if you remember, we went through a set of standard metrics that this group helped us develop, actually. And that surprisingly didn't come up in that group. So it might be something interesting to look at in the future.
Yeah, I would just add that I think DEV has the data because they do encourage lots of ad hoc submissions, reviews. And there's a success. There's lots of response to those solicitations. So I would suspect that there's good data there for analysis that would give some sense of. I think the range of reviews from being non-substantive to substantive is in some ways also captured in that in the ad hoc reviews.
That's great. Just as a quick follow-up, I forgot to mention specifically, it's of course interdisciplinary research. The more interdisciplinary the nature of the research, maybe there's the greater need for ad hocs, and if there's a bias in the system against interdisciplinary research being funded, that's kind of what's driving my question. But I get it. It looks like this is an analysis yet to be done. Thank you.
Suzanne?
So I imagine a lot of the PIs who are funded are using resources like the LTERs and NEON. And one thing we've been talking about on CEOS is the potential of leveraging units like NEON, the many instances of NEON, and LTERs as an opportunity for the surrounding communities to have input into not just the collection of data, but the kinds of questions that are asked. In fact, CEOS is, I think it was the 2019-20 report, may have that wrong, recommended that NSF invest more in the kinds of research that are driven by not just PI interests, but the interests of the communities in which they're working. So a long lead up to a question. I actually have a question, and it is, did you see any evidence that there is indeed community engagement occurring? And if so, was it community engagement just on kind of the receiving end, or were the communities involved in the ideation and kind of the hypothesis development phase as well?
Yeah, so I wasn't part of the subcommittee that actually did that part of the analysis, but I can tell you from a sense I have from working with NEON that there are increasing opportunities for using, this openly accessible NEON data for all kinds of things, and some of those are programs, additional programs being supported by the National Science Foundation. So. I suspect that's happening. There is a response of the community, including in the ideation. What kinds of things, what kind of opportunities they are.
So Suzanne, good morning, everyone. I'm Simon Malcolm. I had a conflicting meeting, which meant I came late. But I'd like to try and respond to your question. I think it's very hard for the COV members to actually be able to tease apart an answer to your question. But what I can say is there's been increased emphasis on being able to engage local communities in not only the research, but also coming up with some of the ideas. We see that in particularly LTER. We're seeing that in the COPE program, which BIO is a partner with, which is run out of the GEO Directorate. And also NEON as well has been actively engaged in being able to bring in diverse communities to be able to address particular questions that can be using those data which are freely available. So I think it's very hard to be able to answer your question from the materials which were the self-study materials that were provided to the COV, but there's certainly a lot of other activities ongoing in that regard. And that's something that we might be able to dig into as well to get a more of a concrete answer.
Gretchen?
Yeah, so I noted that this is sort of a departure, but it ties in a bit. I noticed that there's a disconnect between the number of minority serving institutions receiving grants and the number of minority investigators. And you end with sort of praise for the success of the latter, but I'm worried about the deficit in the former. So the rate of funding for MSI in DEB is lower than the rest of NSF, and I think that is a problem.
Yeah, I agree. It is a problem, and in spite of the increase in number of MSI PIs that was observed, and the fact that there are more MSIs that could be investigators that could be applying for funding, they hadn't really matched the rate of funding overall.
Gretchen, I agree completely. This is work that still needs to be done. And I think we also need to, certainly we're increasing our outreach efforts to engage more minority serving institutions. And there's also, at some level, I think there's less interest, to generalize, there's less interest in ecology and evolution among some minority serving institutions than perhaps there might be in some of the health or molecular kinds of approaches in biology. So I do think there's, we're certainly providing funding to the professional societies to be able to encourage diversity within those societies, and hopefully that will trickle down to an increased number of submissions that we see from minority serving institutions to our programs to be able to diversify the general portfolio that we have within DEB.
If there are no other questions, Kennedy, sorry.
I just want to touch base on one item that Simon just talked about, the idea about the lack of MSIs having those specific type of programs. So when you look at all HBCU and most of the MSIs, they don't have evolutionary or those kind of degree programs for their students. And when you think about this in a larger framework, that means that there's a students from those populations who are not getting exposure to that specific type of field. And so the bigger question is, how do we address this idea about a specific group of institutions where students are not exposed to certain programs at the undergraduate level and not the graduate level?
I think I'm going to ask Bianca to respond to that one. Or Alan. Okay. Yeah. What do you think?
This comes back to, we really have stepped up our thoughts around how we do outreach and how we connect with those institutions, even if they aren't institutions that are submitting to us, but being out there and present and showing the kind of professional opportunities that exist and the value of the diversity of the education. So it is, we're rethinking, not going back to those institutions that we're good at recruiting from and getting grants from, but going to institutions we've never had a grant from and reaching out to them. But yeah, we see that as a problem.
Jim, were you gonna? Yes, Jim.
Yeah, thanks, Kenny. Just to your point, we do have this RCN UBE program that's really, the goal is to form networks of folks trying to transform undergraduate biology education. So at the undergraduate level, that just sounded like a perfect project. that could be submitted to that program to help deal with that curricula availability problem that you highlighted. So I just wanted to mention it.
Suzanne?
Just a quick thought, when it comes to recruiting, the message and the messenger are important. So you might also think about the demographics of the NSF workforce and the degree to which you're positioned to send people who kind of look like the folks you're trying to recruit out into the field.
Okay. That seems like if there are no further questions, a very
So
good.
Simon did call
sorry,
on me.
there are,
So
Bianca.
I wanted to go back to Kennedy's point, and I think one of the most untapped activities that bio supports is the REU. So I went to Xavier, and Xavier has no specific sort of degree components in the DEB space, but all of those students are required to do research. And so I never did research in the molecular space. I actually spent a summer with dolphins doing something so totally different. And so I think connecting those institutions with those opportunities is really the key to get people to get young people to think about science in a different way. So all I ever saw was, chemistry. Sorry, Teresa. Chemistry.
I'm an engineer.
And X-ray crystallography. Sorry, Susan. But but in those summer experiences where it wasn't molecular and biochemical and biomedical, where it was just specific basic biology, that's where I really gained more of appreciation. So I think connecting those institutions with those other opportunities that we're supporting are the key to build that pipeline.
But what area did you finally get your PhD in? It was not in dolphins.
It was in microbiology. I did study chemistry, but I woke up after my summer experiences and left chemistry behind. Sorry, Susan, again.
Oh, thank you.
So I'll speak to what Bianca was saying. I know, for example, in Geo that they have some REU programs where they limit the schools that they bring students in from. So they have REUs that just focus on community colleges, for example, to bring students in who don't have specific disciplines in their community colleges for exactly what you're saying. I don't know if bio does anything like that, but I thought that was a very interesting model they use in Geo. Okay, apparently time has been called. That was a very good discussion. Thank you, everybody. And we'll take a break and we will reconvene at 1 p.m. Susan wants to say something before we reconvene. No, she's going to. Okay. We will reconvene at 1 p.m. Eastern. Thank you.
Paul, is he still on?
From chemistry.
No. He does. No.
Yeah.
Recording in progress.
Okay, welcome back, everyone. As Susan mentioned in her update this morning, a focal point for this meeting is how NSF and BIO are responding to Biden administration priorities as laid out in the Bioeconomy Executive Order, which was signed last September. To begin the discussion, I will turn to Teresa Good, who has led NSF's response to Section 3 of the EO, which called for federal agencies to identify and report on R&D goals in biotechnology and biomanufacturing to further societal goals in key areas to advance the U.S. bioeconomy. Teresa?
Thank you, Mike. Let me pull up my slides. Okay, there we go. Good afternoon, welcome back. I'm going to take the next 10 minutes or so to provide you with some context about NSF's interest and priorities associated with the bioeconomy. And I've got three main messages to convey. Why now? Well, yes, there's an executive order that you've heard about, but there are also scientific and societal motivations behind some of the sense of urgency that we feel at NSF. Who? There are a lot of stakeholders involved across the federal government, across NSF, in the community, industry, academia, and the public, and we need to coordinate. What? In the report that you've read that are of the bold goals, you've likely noted there are a lot of R&D needs to achieve the societal benefits that they outline. And you've heard from Susan about the projected budget. We're going to need to prioritize. So hopefully, as I address those 3 points, you'll see I will tee up the conversation that you'll have with Brent and later in the breakout discussions around coordination and prioritization. And also set you up to think about the other needs beyond research and development needs, such as workforce and infrastructure, that Celeste and Sri will talk about later in the AC meeting. So why now? There's the executive order on advancing biotechnology and biomanufacturing innovation for a sustainable, safe, and secure American bioeconomy. And we'll talk a lot about that in some detail, a bit later. There was also the CHIPS and Science Act of 2022. And in that, there is a national engineering, biology, research and development initiative that calls for agency coordination, as well as NSF-specific support for research, including research into the societal and environmental implications of biotechnology and workforce development. But even without the law, CHIPS and Science, and the executive order, there's a tremendous opportunity and a need for NSF and other agencies to advance the biotechnology to address pressing societal challenges. Advances in the biological sciences and other fields, such as AI and imaging, have advanced to such a point that we can now more than ever understand and deploy living systems. Stop that. To advance science, the economy, and address societal challenges. Those challenges addressing climate change, sustainable food production, improving human health, and ensuring resilient supply chains are critically important. Perhaps some of them are a crisis, and there is no time to waste in deploying solutions. If we don't act now, others will. And we risk falling behind, as we did in semiconductors and telecommunications. If we cannot translate biotech innovation into societal and economic benefits for all Americans, we will lose the economic advantages, but we will also lose access to the data if other actors take over leadership of the bioeconomy. And if we, in the US, are not part of the leadership across the globe in the bioeconomy, then we will not be able to ensure that our values of equity and who participates in and who benefits from the bioeconomy, as well as ethics, environmental stewardship, openness, are part of the global framework for the bioeconomy. So let me start by telling you what's in the executive order. There are a lot of sections in the executive order. The things highlighted in gold on the slide are the things that NSF played a key role in developing the reports associated with them. Specifically R&D needs, data for the bioeconomy, biomanufacturing ecosystem, workforce, and international engagement. The goals of the executive order to really spur federal agency investments in research and development to increase domestic biomanufacturing capacity and to enhance regulatory clarity. Through all of these different activities that are all interconnected, hopefully we will achieve some of those goals. Okay. Clearly, I'm not great at the page turning thing. You were smart. Anyway, so in the executive order, so I'm going to focus now on the section 3 that was really the R&D needs to advance societal goals. In the executive order, different agencies were charged to write different parts of that report that address different sectors of the bioeconomy. HHS, the report on health, or the part of the report on health. The Department of Energy wrote the part of the report on climate change and clean energy, USDA on food and agricultural innovation, commerce and defense on supply chain resilience, and NSF was charged with writing the report on cross-cutting advances that would be foundational to all of that. In writing those reports, there was a lot of stakeholder engagement. That there were roundtables at the White House with academia and industry. There were requests for information that resulted in more than 200 pages of input from the community. There were conversations with the Engineering Biology Research Consortium. We leveraged the workshops that we had held previously that summer with academia, industry, and other government stakeholders. We had listening sessions within NSF to engage in program director expertise. We had different members across the bio leadership and across NSF leadership help draft different sections of those reports. That was our process. Other agencies had very similar processes. Anyway, that's how we got to the reports that you saw. And that in each of the reports, there were multiple themes, anywhere from 3 to 6, that highlight what are the research and development needs in each of those sectors. And what I would tell you is that those themes are interconnected, that if you look at the AI-driven production of therapeutics or advances in cell-based biomanufacturing of cell-based therapeutics, some of those things could easily, that are in the human health report, that foundational research done by NSF could feed into some of those themes. The report from Energy on the theme on climate-focused agricultural systems, as well as the section from the USDA report on improving sustainability and resource conservation in food production. Clearly, there are interconnections and programs like the NSF Plant Genome Research Program or the Centers in Programmable Plants feed into the basic research that feed into those themes. So there are many connections between all of those reports. An NSF task, then seeing all of these themes, was to write a report that would describe the cross-cutting advances we need across all sectors that would enable all of those themes and all of the different societal advances. The other thing that we needed to do is to consider the equities across all of NSF, because at NSF, every directorate and office participates in the bioeconomy. And so these are the 6 themes that NSF came up with. There is leveraging biodiversity across the tree of life. I'm going to turn my page. Give me a second. Sorry about that. Leveraging biodiversity across the tree of life. We have barely tapped into the biodiversity
across the tree of life and the innovations from evolution to develop applications for the bioeconomy. There is tremendous potential yet untapped, and to develop that potential, we need to speed the rate at which we elucidate function of that biodiversity, to develop tools to utilize the existing biological data, infrastructure to mine that data and to synthesize it and to discover, and new tools for discovery and exploitation of diversity across the tree of life. Prediction and design. There's tremendous opportunity to to exploit the advances in AI, especially in mechanism and theory constrained AI, to predict the function of organisms and biological systems, and then to solve the reciprocal or inverse problem of designing based on that predictive knowledge. Build and measure. We've made great strides in building biological circuits from groups of genes in the simplest of organisms. And we are beginning to be able to manufacture organelles. But we have yet to master how to build a complex organism with desired traits, i.e. a synthetic plant. This will be one of the next big challenges in the field. And our capacity to build far exceeds our capacity to measure, whether it is phenotyping an individual organism or phenotyping or looking at the function of multiple organisms in the environment, so that there is tremendous need to expand our capabilities to measure. To do any of that at scale, for it to be commercially important, we need to be able to scale that up. And while the US leads the world in being able to produce things at the nanogram or microliter level, we cannot reliably scale up processes to commercial scale. And so there are real opportunities to combine what we know about biological systems and process engineering. And we now have the computational capabilities to combine those to actually develop the science of scale up. There are many different kinds of manufacturing systems beyond just what you can grow in a fermenter. And so there's a real opportunity to combine biotic and abiotic systems, perhaps their brain computing or other kinds of systems like that. And so there's an opportunity to innovate in the biomanufacturing space. And then in anything we do, that we need to consider how everyone can benefit from the bioeconomy and everyone can participate in the development or co-generation of technologies if the bioeconomy in the US is to reach its full potential. I really just want to draw your attention to all of this is interconnected. That I'm going to start in the middle of the slide. So the third theme I talked about was build and measure. Well, that is part of what I think about is in terms of as a design, build, test, learn cycle. And that is a part of how we do biological design. Well, in order to do that, in order to continue to innovate in that cycle, we need to bring in new function, new discoveries. And that is really how we tap into the biodiversity across the tree of life, that is really essential for us to continue to innovate. That we also have to be able to predict what those new functions will be from that biodiversity, and that feeds into how we also enable design. That for any of those things that we build in that centered and in that design, build, test, learn cycle, we have to be able to then implement that in a commercial application or in some other deployable system, whether we do that in a fermenter scaling up or we do that in other kinds of manufacturing innovations. And all of that has to be captured in, embedded within this framework where we think about ethics, equity, we think about environmental impacts and economic impacts, that we involve people from the very beginning in co-generation of technology. We think about public engagement and risk appetite. So all interconnected. And so with that, hopefully, I've given you a flavor of what is in the Bold Goal Report. And hopefully you will have seen it's urgent. There is a compelling need. We can solve really important problems. That there is a need to coordinate across all of the agencies, across all of the stakeholders to achieve goals in every sector. And that there is a need to prioritize. We cannot do it all at once. And with that, if you don't have any questions, I will hand off to Brent. sharing.
Good afternoon, everybody. Let me get my slides up. 1 second. All right. Good afternoon, everyone. Thank you, Teresa, for the perfect lead into the Casa Bio activity. I'm Brent Miller. I'm the Science Advisor in the Office of the Assistant Director. I'm going to introduce you to the activity we're pulling together called Catalyzing Across Sectors to Advance the Bioeconomy, or Casa Bio. The challenge ahead is, pretty bluntly, the bioeconomy is a huge research and development space with significant potential. And with this challenge, the questions become, how can federal agencies prioritize and stage activity strategically to realize the executive order's bioeconomy? We have an executive order that places the responsibility on multiple agencies to develop all of government plans to reach an ambitious bioeconomy end state across multiple sectors. And to remind you, those sectors are climate change, food and agriculture, supply chain resilience, and human health. Ideally, we think if the stakeholders approach this together, there is real potential to place the U.S. bioeconomy on sure footing. In reality, the EO vision exceeds individual federal government agencies' resources. And the question becomes, what do forward-looking research paths in the executive order's R&D space look like? And part of that answer to that question was the Bold Goals report that Teresa just introduced you to. And as a reminder, it included the federal agencies on the right in the slide, and it included input from relevant stakeholders through recent reports and workshops, direct consultation with subject matter experts, listening sessions, roundtable convenings, and an OSTP-led request for information. But now we're moving beyond the Bold Goals. We need to hear from a broader set of community as stakeholders that includes federal, industry, and nonprofit stakeholders, and the research community as stakeholders, with the goal of finding aligned areas of interest and synergistic ways forward. This is the goal of Casa Bio: bring alignment between the various stakeholders, both funder stakeholders and the research community stakeholders, to identify synergistic paths forward. So this is a bit of a spoiler alert. I'm going to start at the end. That is what we hope to achieve with this activity. Starting on the left, the three things that we hope to achieve with Casa Bio. First is generate research community excitement and nucleate like-minded groups. Second is establish roadmaps that represent how different agencies' goals link together, how research communities would approach the pathway, and what results and impacts may be expected by following the roadmaps. And third and finally, an understanding of which roadmaps have multiple agency interests. As the slide depicts, we envision multiple roadmaps for consideration by the stakeholders involved. What do we envision in these roadmaps? Listed here are a few ideas. We're still in the planning stages, so this list may change, but whatever the list is, a goal for the roadmap is to demonstrate the potential for synergy and impact on the bioeconomy R&D space and future translation. And we want the roadmaps to be actionable for the agencies involved. So the question I hope you have now at this point is, how do we get here? This approach is spelled out in multiple steps, and the first step is alignment. This is where we pull together funders to align their interest and create a call to action that represents a unified ask to the research community. And again, these funders include government, industry, nonprofit, or philanthropies. The call to action would focus on the areas of interest that funders have identified, which have synergistic or collaborative potential, or another way of putting it, areas that have multi-agency interests. The call to action may look something like, agency X, Y, and Z are interested in an area like enhancing predictive modeling and engineering design of biological systems. And the call to action asks the research community to provide their big questions or their blue sky projects that may be relevant to this space. And additionally, give us your thoughts on the associated expertise needed to address the big question or blue sky project that you've put forward. Step 2 is engagement. This is where we engage the community with that call to action and solicit big questions or blue sky projects relevant to each area of interest. The goal here is to have many brief responses that align with the areas of interest identified in the funder group in that alignment step. This is really the step where the community has the opportunity to show their enthusiasm for a specific area of interest. The next step is a synthesis step. We'll reconvene the funders and work to synthesize and characterize feedback from the research community to better understand its fit to the administration or agency interest and capacity. The goal at this step is for the funders to gain a deeper understanding of the research community's interests and capacity and identify priority goal areas that rise above the others. That is, we're looking to narrow down the field of areas of interest identified in that alignment stage. We'll do that through quantifying the community's enthusiasm for that space, or maybe look at the nature of the big questions and blue sky projects in that space. The fourth step is advancement. This is where we focus on the research community's energies on those narrowed down priority goal areas and start putting more flesh on the bones. The intended output from the research community in the advancement stage is documented views of how they envision implementation of individual pathways. That is, those roadmaps that I presented in the previous slide. Our hope is that these roadmaps will be broadly publicly available, potentially via publication in professional journals, and be actionable by the funders we've gathered for the alignment stage. This gives you a timeline of phase one, which includes alignment, engagement, and synthesis steps. August and September, where we're at now, we'll be planning and coordinating with No Innovation, who will be facilitating the Casa Bio activity. As well, in August and September, we hope to have our 50 to 60 funder representatives complete. And the characteristics of those folks were covered in the one-pager that you received in your AC packet. So far, we have 40 federal participants confirmed from 16 different organizations and sub-organizations. Those include Centers for Disease Control, Department of Commerce, Department of Defense, Department of Energy, EPA, Food and Drug Administration, NASA, NIH, the State Department, USDA, HHS, and even the Department of Justice. We've recently asked each of these organizations to suggest industry and philanthropy partners that they would like to include in this conversation, and some of these individuals be added to our roster of participants. So the week of October 23rd is when our alignment meetings will happen. We'll pull the stakeholders together to align interest and create that call to action. And this will happen via a quick 2-hour orientation with funder decision makers and those subject matter experts. And this is to set the stage and generate excitement and guidance for the next longer meeting. That next longer meeting is the 2-day in-person alignment meeting with subject matter experts. And this will likely only need the subject matter experts, leaving the agency organization decision makers free to participate if they want. In November and January, this is when we hope to engage the research community. We imagine here 4 town hall style meetings of 90 to 120 minutes each, and these are facilitated virtual meetings with 500 plus individuals apiece. And finally, in January, this is when the funder representatives will reconvene to synthesize the feedback from the research community. These are short virtual meetings spread out over multiple weeks to lessen the burden on our participants. On the left is how we envision the output of phase one, synthesis sessions. It represents a few things we may want to get from the synthesis steps about each specific priority goal area. This will likely evolve as more detailed planning commences, but we want to define the priority goal areas in these ways. Obviously, first, identify the priority goal area funders want to represent as a meeting. What are the big questions or blue sky project themes addressed by this priority goal area? What research community expertise is needed at the meeting's activity? What agencies or organizations are committed to participate in the priority goal areas? The right side of the screen represents multiple meetings that will form the output of the synthesis activity. The idea is that there's one meeting for each priority goal area. The meetings will be facilitated, maybe in person or virtual. We're still deciding that. They will consist of less than 100 people each, and they will last 3 full days. It is the output of the phase 2 activities that we intend to be actionable for the funder stakeholder group. This is where we build our roadmaps. And as I explained at the start of the presentation, we hope to have engaged and excited research communities, multiple roadmaps that show direction and potential of R&D within the defined areas, and a better understanding on how all the stakeholders can participate better together in those defined areas. So 2 things before I stop. First, thank you all for your attention, and I'm happy to field questions with my colleagues. And second, I'd especially like to thank Karen Cohn and Teresa Good for being willing and active partners and bringing this activity to this point. Their deep knowledge of the bioeconomy space is really invaluable. I'll stop and take questions.
All right, thank you both for your presentations. So we've now got a few minutes to take questions for both Brent and Teresa on their presentations. Hank.
So this is all very exciting. What I'm thinking about is the cephalopod that edits its own RNA that Susan talked about earlier. But what's boggling my mind is, how little we know about all the things organisms are capable of, and how, an effort like this is going to generate that kind of knowledge. Because I think in that kind of just really basic, exploratory research, we're going to learn things that are going to be helpful for the bioeconomy. But it's just so much of it yet to discover.
No, I think that we all agree, and that is part of why that first theme in the NSS report is how do we leverage biodiversity across the tree of life, that it's through that discovery, and I don't think that anyone thinks we're done with discovery. And whether we do that discovery strategically or we do it broadly, I think that there has to be all of that, and that has to be the foundation upon which we build all of the other innovations. I think we need new tools to help us accelerate the discovery. And I think if you want to get Simon going off on the interwoven data fabric of the tree of life that you're going to need across the bioeconomy, that's one of his favorite topics. But I think that we agree with you.
I just want to comment. You bring up a really good point for all of this, is that it's easy to articulate what we know, and it's difficult to articulate what we don't know. And so it gives the false impression that we have more of an emphasis on what we know and so I think the onus is on us as we go forward to make sure that we don't get trapped into exactly what I think you were worrying about behind that question, is because you can't, right? We don't have a crystal ball. We don't know. And so we can't outline it in such precise terms that we can outline the rest of it. And so keep asking us those questions to make sure we don't fall into that trap of doing it. Thank you.
Don't worry, the report's going to be called The Unknown Unknowns, to quote the famous man himself. All right, next, Tom.
Actually, really quick response. Also, Hank, an important aspect of this is trying to understand what the pull is. And that's why we're bringing the industry aspect and those folks in, so that we understand what the pull is and we know where to strategically do the discovery, hopefully. I mean, it's sort of trying to understand what those known unknowns are. What is the need? That's the other aspect of this.
All right, Tom.
Yeah, this is probably a sort of a pervasive issue throughout all of this, is what is your anticipated role, particularly in these early steps of engagement that you're doing and alignment with sort of both public and private strengths in AI, and also critically the unfortunately largely private strengths in AI infrastructure, largely compute, because that's going to be core to so much of where you're going to go.
I'll let you run at it first.
So I think that is part of what we're working through now. I think that one of the reports that has yet to be released is this report on data for the bioeconomy. And a lot of that is really how do we, so there's what does that computational data fabric look like. But I also think that we are just beginning to explore how do we bring in that expertise. And our partners in SAIS
are going to help with that. I think that as you talk to Sri later and ask him about infrastructure in particular, I think that he may have some answers there. But I think it is a really great question, because I think we don't know how we're going yet. How we're going to really tap into that. And I don't know if anybody wants to save me there.
Yeah, so
could I just add one more little twist to that, Teresa? Thanks, that was great. I think this is the concern I'm having, is that the access to high-end compute is getting constrained. And I think we're at a stage where it's more like the federal highway system, that there needs to be something where there's open access broadly. And this is going to be core to all of NSF's successes in frankly, multiple directorates. That there can be elevation of that. It's absolutely critical for bio, but it's absolutely critical broadly. And it accessible is the key worry I have.
And Tom, I'll add to what Teresa was saying and hopefully address some of the thoughts you're having. In the CASA bio activity, one thing that we're trying to figure out how to inject into it, as Teresa said, the data for the bioeconomy report isn't out yet, but we want to make the data for the bioeconomy a real aspect of this. So we want that on people's front burner when they're asking the questions or trying to get to the call to action from the community. So it will be part of this conversation. I'm imagining that the report for the data for the bioeconomy will have its own unique approach to getting what they need, but we're definitely going to bring it into what we're doing, at least part of the conversation.
And one last thing, that certainly in the background, as we are planning and thinking about priorities within bio, that there are lots of conversations that are happening between bio leadership and the size leadership around this topic. And while I'm not privy to all of those conversations, just know that lots of them are happening.
Sorry, this is Sri, can I add a couple of things? I'm sorry if I'm speaking out of turn, but so there are several programs that NSF has stood up for access to cloud and other high-end compute, particularly for AI. I mean, we've always had a very large high-performance computing access program through Exceed, as well as the supercomputing centers. But for instance, Cloud Bank and several other programs are being stood up, and there is constant conversation about how to make this access more broad and pervasive. And you will see later on when I present a little bit about AI institutes and the infrastructure that's available, there are conversations sort of broadening this and bringing industry partners into this. And there is recognition across the foundation that access to compute is becoming a large bottleneck. And we really need to resolve this, not just for bio, but certainly for bio, but for many other fields as well. So thanks.
Maria? Can you unmute, Maria?
Finally, sorry. My question is about the translational and applied aspects of the larger project, and how much of a role that NSF is anticipating to play in putting in funding and putting forward translational, again, applied aspects of basic science.
I'll start and then I'll hand off. That while there's some things that NSF Bio is doing, we have a partnership with TIP with Noble Reach Emerge. I may hand off to Brent to tell you more about that, where there are, in this pilot, there are multiple, we have commercialization experts embedded with our PIs to help them translate their projects, and that's ongoing. There are other parts of NSF that are much more engaged in translation, particularly the Technology Innovation and Partnerships Directorate, though their translation activities, whether they're engines or SBIR or the like, are sector agnostic, but many of them are in the bioeconomy, and there are activities or partnerships that we are developing with them and with others. So certainly some. I don't know what you want. Go ahead, Ed.
The only thing I would add, I think, is the conversations for Casa Bio are really not, we're trying to really avoid the budget question, to be really honest. We're staying away from that because budgets are uncertain. What we're really trying to get at is to try to find those scientific spaces where there is synergy between the groups and then isolate those and then put those out there for a broad audience to see and respond to. And I can't imagine who all of those broad audiences are, but we hope that a lot of people see the output and that it's actionable and they see the value of it in us approaching it those ways.
All right. Suzanne and then Gretchen.
Thanks. I agree. I think this is really exciting, and I love the idea of kind of coming to consensus on what needs to be done. I hope in the process of having those conversations, there'll also be a conversation about where it needs to be done. You know, one of the things we've talked about before, and it's certainly something that comes up a lot on CEOS, is the idea that there's the potential to empower smaller institutions, minority-serving institutions, to participate in ways that they maybe haven't participated in the past. You know, up until now, I think the tendency has been for a smaller institution to be the stepchild, so to speak, the lesser of the entities when it comes to doing these important projects. And, you know, perhaps there's an opportunity now, particularly with the focus on computation and predictive modeling, to empower the kinds of institutions that perhaps don't have robust research infrastructure in a way that will not only allow them to contribute to the kinds of things that Casa Bio wants to achieve, but at the same time develop that workforce that we were talking about earlier today. So I just hope we'll think not just about what's to be done, but where it's to be done as well.
So thanks for that comment, Suzanne. I think one of the things that Sri will talk about when he talks about infrastructure in particular is what are we trying to do around kind of democratizing access, and whether that is in biofoundries or in other programs, but that democratizing access to the tools to participate in the bioeconomy as well as to benefit from the bioeconomy are part and parcel of what we are thinking about in these programs. It's part of what's happening through the Engines program. It's one of the review criteria there. So I think it's something we think about. Also, I think that one of the agency priority goals are around increasing funding in EPSCoR states, and that is always prevalent in our minds as we are going forward. I don't know if, Brent, you wanted to.
What I was going to say with respect to the Casa Bio activity, I think we're going to try really hard to make sure that those call to action meetings and the later synthesis meetings are going to be wide open for everybody to participate in. There's going to be no restrictions there, and they're large meetings, and we're hoping that we can get the word out sufficiently, not only through ourselves, but through our partners that are going to be involved in this, so they reach out to their communities and make sure that everybody knows that this is happening and everyone has a chance to contribute.
Okay, we've got time for one. Gretchen, do you still want a quick question? Yeah,
thanks. So I noticed in your discussion, Teresa, of the biodiversity across the Tree of Life, it seemed like you were sort of treating biodiversity as a collection of organisms, all with special functions, most of them unknown, that have yet to be tapped. And I think that there are a lot of functions of biodiversity that are known. And I think that this may be one of the reasons that DV doesn't get funded so much, or doesn't attract students who want a good living, is because we don't pay for the kind of services and effects that biodiversity actually rewards. So for instance, just protection of coastal communities and the kind of economics of preventing environmental disaster. So I think that the biodiversity of communities is also important and should really be something that's examined.
Just as well that I was on mute. I admit that the focus has been on how we can leverage biodiversity for economic benefit and not the economic
cost of losing biodiversity and or the kind of real value in conservation and other activities. This is so outside of my field. I am going to hand off to anyone who will take the floor. And I'm looking at perhaps Alan. There we go. Thanks, Alan.
So one of the nice things about being at NSF is the diversity of people contributing to these conversations. And Teresa's reminded on a regular basis about ecosystem services, and variation is a key. And so part of my job is to gently remind her that engineers think everything works in one way, and we think it works in many different ways. So it's fun, right? We have a way of being inclusive internally as well as externally, and we recognize that's an important component. So just a reassurance that this is not being ignored. It's easy. I think the comment of it's easy to talk about what we know is an important one. We try to keep that in the back of our mind as the things we're not familiar with are important too.
And I just wanted to add, there was one conversation that I was having with Mike while we were having lunch, and I think that was one of the things that's really important about what we have been doing in this space is we all recognize what we contribute and where we have no business contributing, and we need to call on our colleagues to contribute. And so while I may have provided the presentation, this has really been an effort across all of NSF. And I had forgotten when I was talking about it, that really the primary authors or the final authors of that report that you read, the NSF part of it. So I really have to thank Karen Cohn and Jared Dashoff, who are not here, who did really a tremendous amount of work on that report.
All right. So thank you for the presentations, Teresa and Brent. Those were great, and for the good discussion. This is a great segue to our next activity. So we're now going to take a small break. You should all do you all know who your breakout group is? It's on the agenda. So what's going to happen is the next session, the AC members, we're going to discuss priority research areas that could advance the bioeconomy. So it's a great segue from what we were just discussing. So think about the reading assignment, which I know you've all spent all night studying, the guiding questions we were given, and what we have heard today. So just to remind you, members of the public have the option of staying on this YouTube channel to hear discussions from the group that will be in this room, or there will be new YouTube links to the other two hybrid breakout groups also. So also bio staff will help with the Zoom setup and the meetings in the other rooms. And then after the session, we'll take a 15-minute break and we'll resume at 3 o'clock for a report out from each of the breakout groups. So does anybody have any questions about how that's going to work? All right, look at that.
Let's see.
Just like when I asked my students that in class, like, yeah, it's all good. All right, see you all soon.
For those of you who are leaving this room, the breakout rooms are just out this door and on the right. And Kim is going to be in one of them, and Brent's going to be in the other, and I'm staying here.
I go to the
Senior
other room.
managers are free to stay or watch from your office. And Philip, can we have that slide that shows the
Page 4.
Right?
Yeah.
No.
Yes.
Yes.
You got it?
I got it. Melody sent it to me.
Log out.
I could have stayed here.
Oh, really?
There would be an extra.
There will be a report of it.
Okay, so that's good.
Yeah.
No, I can.
Charles.
I don't need it for plugging in.
Oops, there it goes there.
Eric, you need to change rooms.
Figure out where I'm going.
Am I in the right place? This is Suzanne.
Suzanne, you're not in the right place. You should. So, Suzanne, you should be in room group 2.
Okay. My bad.
Eric, you should be in group 3.
I think I'm in the right place.
You're in the right place, Paul.
Okay.
So, Paul and Maria, stay here. Don't go.
Folks, can you hear me?
You're in the wrong room, Tom.
Yes, I know. I went to room 3 and it's only a webinar link with no video or sound. The link at the bottom of the Word document. So I went, the Karen sent the word.
It'll get started in a second, Tom, she said.
Oh, it was started, but they can't hear me or see me.
Okay.
I'll go back to it, but just letting you know, I can hear you.
You'll get technical help. Suzanne, you need to be in group 2.
Okay. Leaving.
Remote participants signing in. I don't know if our screen is set up right to receive them.
You might want to take your computer with you, I think, Hank. I'm not sure.
Maybe the way to do it.
Yeah.
Okay, good idea. Guess I should see them in here right now, right?
Scott should have taken his to. Yeah, because otherwise you're not going to show up to the YouTube participants.
Okay, I got it.
I need to go and tell Scott to do the same. Scott, the last one left here. Just our usual business. Yeah, but what I don't understand is what. Not very. Okay. So, what I suggest you do is minimize the shared screen component, because that's going to stay up, because I think that's if people come and look for the YouTube link for public access. And I think, Hank, you have to log out of this Zoom. I'm going to go and tell Scott to get his computer as well.
Hank, can you hear me?
You're in the wrong room. Hey, Rob, you're supposed to be in a different room.
I know.
Hank,
I used the link in that. I used the link given in the agenda, and it got me into the room. I could see Hank, but I was in a view-only webinar type space. So, and I presume that happened to the other people who clicked on that link that's in the day one agenda breakout group 3.
I think that Karen said just go there and it will. She'll change it.
Okay. Okay, I'll go back there.
Okay, if anyone has any energy left to discuss the big questions of the day, after all of that, our task is I think basically to discuss priority research areas that could advance the bioeconomy. So who would like to? And this is a blessing of being the chair of BioAC. I can't be the one that reports out because I have to moderate. So we need someone who will be, we need a note taker and a presenter. All right, Barbara will take notes of our discussion. And who would like to report out? Maria, is that a volunteer hand that you put up?
No, I figured that I should put it down. But I was going to say, can we get rid of the screen, that has the meeting information? And I also wanted to say something about the questions, but
sure.
If nobody else is volunteer, I'll be happy to report out. As long as I can get the notes from Barbara.
Yeah. All right, so Maria, you go ahead. I think it's good. We can just start.
So, yeah, I mean, reading the reports, particularly the EO and then even the CASA report, as an ecologist, I just felt like there's a lot of emphasis on biotechnology, just building critters and building processes and a sort of complete bypass of we have come for, I'm talking about climate change in particular, but we have come to call, nature-based solutions. And there's been in the last month, 2 papers that have come out, high-profile papers, sort of looking at the use that corporations are making of carbon markets, and particularly this claim of carbon neutrality, and how it really has absolutely no evidence that there's having any impact on sort of carbon. And that has many connections to industry and how they are going to meet this kind of plan. But also, the real difficulty which is monitoring and bringing in experts and all that. And there was zero of that. Everything was just biotechnology based, rather than monitor. There was no monitoring in the climate change angle or technologies to monitor and so on. And that's a huge market. That is.
I was going to say, that I really appreciate what you're saying, because to my mind, what is biotech? What is the market for biotech? What is the bioeconomy? I mean, mitigating climate change surely is the impact on every other aspect of the economy and of modern life seems to, I would think that that's fairly important. And just as an aside, when I looked at I don't know if you looked at this one, this is from the DOE, they're harnessing goals for biotechnology and biomanufacturing. Some of this just seemed to be slowing the inevitable rather than looking for new ways, better aviation fuels, things like that. I thought that's not really the point, is it? At this point.
And there was an article in the Times a couple of days ago about all these carbon capture technologies, which are nowhere near where they would be. And there's necessary research to be able to figure out how we're going to remove carbon from the atmosphere and put it somewhere. But in the meantime, the cheapest solution, and the most obvious solution, and probably the least damaging, is monitoring what is happening, with forests, what is happening with soils, and so on. And the business world has jumped like crazy on this, except that they are not doing what they're claiming to do. And so, I just feel it's a glaring hole in the whole battle.
Can I make a comment about this?
Yeah, please.
This is a great opening thought because, full disclosure, I work in biotechnology, I have a foot in industry, and things like drug discovery are part of what I do, where it's kind of like this, it's got this drumbeat of an ethical responsibility because we all care about our health and we want to save human lives. But, we're not feeling this ethical responsibility about the earth, right? And, I'm listening to Maria's comment. It's like, this is like spot on that, if you don't understand the thing it is you're trying to preserve and save, then, how do you really leverage the bioeconomy to be better to go about saving it? And it's, I don't know what to call it, missed opportunity or just, short-sighted to not kind of have these go hand in hand. So I don't have any great solutions beyond that, but I think that this at the beginning is a very good comment about what's being missed. So thank you for saying. It's a total conversation stopper. Sorry.
No, but to your point, Paul, it kind of links almost. If you're doing human health, like you say, this is human health also. There's a lot of this that impacts human health. The healthy living strategies we see, new drug development, that's been shaped by climate change as well. It's not, the way that diet is shaped by climate change. The things that everything is, it's that interconnectivity which we probably poorly understand. Is that within NSF's scope to look at this part because it impacts bioeconomy? It's a basic research area.
Yeah, I feel like the whole One Health perspective that has been gaining a lot of attention is a valid way to think about interconnected systems. And it's very true. You can't really look at one in isolation. So I found that there wasn't much of that when I was reading this. I was surprised how little of that, because I have undergrads coming up to me who are, they're perfectly intelligent. I'm not saying they don't know how the world works. They're learning, but they'll remind me, hey, oh yeah, your group looks at something and here's how it relates to One Health approach. I'm like, oh, okay. Well, I guess they're reading the news or paying attention. So it's Karen, kind of surprising. Sorry, I'll be quiet.
Karen wants to add something quickly.
Sorry, I'm not really supposed to be participating, but I do have an answer for you for why there's not a One Health focus here. And that is that when we were trying to come up with the strategies for writing these reports, all of the agencies were collaborating with each other to try and figure out who was going to be responsible for which report, what was going to be in those reports. In some cases, so if there's not a One Health aspect to anything, it's because none of the agencies really got the buy-in that they needed to put that in their report. And also, Paul, we supported the Bioeconomy Coordinating Committee at NSF supported a workshop about a year and a half ago. UIDP sponsored it for us. there was a workshop on One Health, so we have had a lot of community input on that topic. And I'm assuming that we'll hear that topic surface again when we get the community back together, because I know that there's a lot of interest in that particular topic. For NSF, that topic is spread across, support for that kind of topic is spread across a lot of the directorates. But so that's why there's just not, we didn't end up with the reports addressing every single thing that could have been addressed. So there had to be some lines drawn. We were being given very heavy direction by OSTP for what was in the reports and how to formulate them. And so I know that some things just didn't show up that might have been favorites of some people.
So, sorry, Maria, I just saw your hand went up again.
I just want to sort of emphasize that, to me, it seems like I don't think there's any part of the federal government that is addressing this issue. Certainly not NASA, which would be the most natural partner, in terms of kind of monitoring what's going on with Carbon Partners, because they don't have, the same breadth that NSF has. I just saw a questionnaire come through a colleague of mine. The Bezos Fund is really interested in this. But what they're going to do with this, who knows? They're interested in the application of AI to sort of monitor, any kind of nature-based solution. But again, I just want to emphasize that there's tremendous interest in the corporations about this, simply because it's a way of attaching some kind of badge of respectability. I mean, I don't want to be cynical about this. I think they're trying to do the right thing. They just don't know how. And it's a really missed opportunity to not have this in there. I mean, I just worry that, the report to me reads a little bit of, with manufacturing and tech-based solutions, they're satisfying, right? Because they're doing something, they're creating something that has a clear impact, right? So this is what we know. And then there's a whole vast space of stuff that is out there that is perhaps just monitoring or trying to figure out what's going on, which we can control, which is usually very messy from a social and political perspective. And it also refers to things like One Health. How do you get into that city plan and where people build and so on, right? And where they move. And it's just more important. So it's a little bit of like the joke of the drunk, looking, beyond the lamppost, because that's what we can see. And I don't think that is the most visionary approach to this topic.
I was trying to think of a way to sort of summarize this. So my take, because, I'm trying to be positive, which I find very difficult around this topic. It seems that really it's about, we need more basic research to better monitor, to better understand climate change. Because it's a false idea that we know everything now and that's it. And there's plenty of bioeconomy around climate change study, around mitigation, and there's primary benefits in that part of the private sector can grow the primary group, but then there's the secondary benefits. I mean, some companies have looked at this. I think is it FedEx that wants to move to all electrical vehicles because it's simply gonna work better for them, and it's about how this is where, my politics are fairly obvious. You have to have enough research to overcome the lobby that doesn't want this to happen. There is enough research to get past the vested interest. We can't put that in our report because Karen will then say that it's not within our space, because there are bigger issues. I accept that. There are lots of different interest groups, and I think to call them interest groups is to do a disservice to the people that work in those sectors. But I think it's about being better equipped to understand climate change, understand what we can do about it, and how it has this domino effects on other parts of the bioeconomy. I don't think there's any.
If I may interrupt you, Michael. I mean,
Yeah, sure.
What I'm trying to say is that there's a whole part of research that has to do with monitoring. just what is going on, from my interest and what I think about every day, just what is happening in forests, right? And all companies are saying you go to book your flight and it says, we're mitigating carbon, we're gonna be carbon neutral by 2034. The reality of it is that this is not true. This paper that just came out, it came out in Science, like 500 companies saying this is happening, and this is what we claim our credits are, and those credits are nowhere near where they claim they are. And it's a technical limitation in many cases of what we do and how we do.
How do we make that into a basic research question and not a policy issue?
I don't think it's a policy issue, because I think that there's a lot of monitoring questions that have not been addressed. There's a lot of startups in this world. I have a couple of students that went into this world trying to figure out, how do we do this? How do we monitor carbon in an effective manner? What is a value credit and so on. I mean, it seems very simple, but it's a big, and the same thing with some of the questions that relate to health, right? We can do this with technology. What is happening, for instance, with suburbanization and the movement of vectors, when you talk about vector-borne disease and so on from one part to another, right? Again, these are basic questions of wildlife-human interactions. They are not about, creating a lot of technology for creating this disease.
So do you think, I just say this because of a project I'm part of. I'm trying to think of ways through this where there are unexplored areas. I've seen in a project that I'm on is that how little, relatively speaking, there is of understanding biotic and abiotic interactions in complex systems. That I've seen that to be I'm such a microbiologist, I assume that everything in a soil system is driven by microbes. And to not understand what the geochemist is telling me about the abiotic complex systems interactions. I know to an ecologist it's obvious, but to some of us this is not, and I feel that it's sort of at the border of different people's specialties that we don't get into some of this. And maybe that's an area of basic research that we're missing, because it plays into Teresa's talk about exploring biodiversity. You're also looking at systems diversity, and both are evolving at rapid rates because of unprecedented change in the systems.
Mm-hmm. Yeah.
Yeah, if I could jump in, I didn't see another hand up. I mean, I agree with what you're saying. I mean, I've often gone on the record, and it's a little dicey to say that community ecology and community evolution need to be spearheaded and gain more information quickly. I'm trying not to say that, I mean, we have gained so much knowledge of it, but the point is it's hard stuff to study. So therefore, it doesn't surprise me we haven't made as much progress in those areas, because people don't really go after difficult problems. And this concerns me, that if you want to improve a system, or even take from a system with bioinspiration to solve problems, yeah, it's kind of a big deal. The problem is just not sexy enough, apparently, to study community ecology and evolution. I mean, I don't get it, but from everything, microbes, I mean, it's just so obvious that we lack information. I don't know how that gets built in to something like this, but I think many of us, this is kind of a no-brainer, that it would be better if we had more emphasis on it.
Right. I mean, even like for instance, I was reading the USDA part on productivity, right? There's been tons of research, sort of looking at the relationship between productivity and what is happening around wherever it is that you plant these crops. Right? There was no mention whatsoever, again, nature-based solution. It was about creating a particular plant that behaves in a synthetic plant. That is what we represented with this. Nothing wrong with that, but there are other ways also that have been studied that frankly, I also think that it's a big part of what DE does. That's what DE does. The science of environmental biology is, what do we get from ecosystems, and how can that be studied to increase productivity, to protect from disease, to mitigate climate, and so on. And all of that, this monitoring seems to fall under basic research rather than a valid approach to the bioeconomy.
It seems to me that a lot of this is what I'm going to call discovery science. In other words, just figuring out what the heck is going on right now. I mean, that octopus study was awesome. You know, in other words, what are organisms doing right now at the microbial level, at the, you know, large mammal level to adapt to climate change? And they are adapting constantly. And if we just took a look, I mean, so to me, you don't need a hypothesis. You just need to go and observe. I mean, I think that's partly what you were saying earlier, Maria.
Well, I think there's also a political issue here, but I think if we group these things into the bioeconomies, we call some things bioeconomy, and we call other things discovery. And, you know, the funding decisions about what directives to fund at what level of funding, all in the bioeconomy side, whatever reason it is. That means, in my mind, that there's less money for so what we're calling discovery science. And in fact, it was interesting in reading the COB report that one of the findings was that these interdisciplinary calls are getting more money than the basic discovery. And I worry that by calling this discovery and calling this other thing bioeconomy, this will diverge the funding. With less funding going to basic discovery and more funding going to this bioeconomy.
So if you call that the biotic-abiotic interface, and then to what Paul said, I think is so true. It's a difficult thing. So this kind of strikes me that, I think that Michael Lynch's minimal evolving cell is amazing, but there are microbes of the same, just as efficient with 10 times as many genes, and we don't know what 70% of the genes do, but they're there. It would be good to know what they do. I mean, it's in a sense that we did the easy stuff with the defined systems, and I'm guilty of this because now I'm in a complex ecology project. I'm called a minimalist, a reductionist, because that's how I'm seen, because I make everything seem so straightforward. But if you think about when you look in those systems, what do all the other genes do? Nobody knows. When Maria brought up, I think at the last meeting, like the tech, we assume that proteomics is so advanced. When we look at a metaproteome, we're looking at a data set now, which is supposed to be cutting edge in a complex system. We can match 10% of the proteomics hits to the transcriptome. And this is considered pretty good in a highly complex system. To me, that kind of sucks. I mean, like, there's 90% of the proteins that there's so much we don't know that, and this could have limitless impact in ways that I liked what Susan said. We don't have to know what it does now. That's not the point of discovery-based science. I mean, CRISPR-Cas9 came out of a yogurt company in Spain. Biodetergents that everybody got into came out of microbial lipases. Like, these things just keep on, you can't say where they're going to come from. The fact that
sorry to interrupt, but can we just kind of label this and just say, in the carbon world, there's a very clear label that I think applies to all of these.
The what world? Sorry, Maria?
In the carbon world, right?
Carbon world, yeah.
There is, nature-based solutions, right? These are not manufacturing-based solutions. These are nature-based solutions. What is already in nature that we can use to essentially. And I think, I think Gretchen brought this point, that we're thinking about this as additive, right? Rather than thinking, what happens if we don't understand this? If we don't understand the value of nature, right? Then it has a damaging effect which also contributes to the bioeconomy in a negative way, right? Because look at the losses, for instance, that we're having from fires, from floods, from hurricanes, and so on, right? So that's also the bioeconomy in a very different way.
I mean, somehow these comments are reminding me a bit of algae, biofuels from algae. We're seeing as this wonderful direction to go, and it worked pretty well in a lab at small scale. And then, it was, I think, profoundly naive to think that, okay, we're just going to create big vats in the desert, and it completely discounted, essentially a community effect. Of course, they're going to get contaminated by other microbes that are happy to be living in that vat, and it's going to ruin the production. So that I often hold that up as like, really? Somebody thought that was going to work through that approach? But I think that's a granular example of, to me, that is a community effect. It's discounting the fact that this biotic-abiotic interface, that it would work when you take it and try and translate it to the thing that had to scale up to make it even useful in terms of production. So in a sense that, I don't know that we would have, I'll just stop the statement there, because I think that that's a fair example. Doesn't have to go in the breakout reporting back, but I think that that's a fair example.
Barbara.
Yeah, so I'm just wondering, and since I'm supposed to be the note taker, partly, and partly to try to synthesize some of this, just wondering if, as a big question, and we're saying what opportunities can be leveraged in this first sort of issue to advance research areas, is just to continue with the discovery science in all of these different areas, that would support biotechnology very directly. I mean, is that what you're getting at? I'm a little lost in the discussion, in the details of the examples, and trying to.
I think. I mean, I just reading the report really raised a lot of questions for me, and primarily was the focus on the sort of biotechnology from the point of view of manufacturing things, right? Things that are artificial, or they have an artificial component in some. And both from the agriculture perspective, from the climate change, the emphasis was on carbon capture technologies, primarily. And that's all well and good. We need everything. We need all of these approaches, right?
Yeah.
But that essentially makes the assumption that other solutions, like nature-based solutions, where we're, for instance, using, forests or using what we know about soils, or building knowledge on how we can monitor forests and soils better, so that we have a better sense of what is the size of this thing, right? And how can we prevent its destruction? Or if somebody buys a piece of land, a company invests in a piece of land, how can we know what they're actually saying, claiming to receiving credits is actually true? Or, when you come to disease, how do we better understand how disease is transmitted, in the interface of human and natural systems? That contributes to the bioeconomy, not from the point of view of sales, although the carbon market is very much an active one, but from the point of view of preventing losses, right? Losses that we're already experiencing in terms of, look at the impact of COVID or any kind of zoonotic disease. Climate change is an ongoing thing every day, right? So that is somehow not considered part of the bioeconomy, and I just take issue with that focus. And I worry that NSF is going to invest more money in these sort of less messy approaches, missing the sort of elephant in the room.
I guess I could shift the topic slightly, so that if we presume that this will go forward and NSF will have this role and some of the things that were outlined, it was brought up briefly earlier, who would contribute to this? And so the thing that I have seen so far in some biotechnology is that the manufacturing to do it, it's not like the facilities are everywhere. Even at very large research intense universities, there may not be the kinds of infrastructure things, again, I think more about the drug world, so small molecule drugs, and we're making phages, and if there's an insistence about purity in making something to solve a problem that is biologically based, you need a manufacturing facility to do it in a pure way. And the point is, I don't know how to get all of our fellow scientists engaged in this, no matter where they reside. So one way is to somehow, when I was reading the LTER report again, I was like, well, if we set up geographically multiple manufacturing sites that people could participate in this kind of entrepreneurial exercise or trying to use bio-inspired solutions, there's got to be an affordable way so that the small consumer is not squeezed out. That's the thing. I feel like a lot of people have entrepreneurial ambitions, but they simply can't get it done at their institutions. So this is going to cost a lot of money, and I realize that, but that to me is the kind of infrastructure that the USA needs, is to have this be more equitable, have people who want to try things out, can they do it affordably?
Karen told me that Shree's going to, it's a really good point. So Karen, I just checked with her. Shree's going to talk about this because the BioFoundries is supposed to start this, but it just scratches at the surface of this. Okay. And before we go on to yes.
Sorry, I'll just say one other thing that is a real focus area for the TIP Directorate with their regional engines program is exactly what you're saying, is trying to put a nationwide focus on being able to enable people to work in these use-inspired research spaces all over the country, so that their new awards are just being announced now. I think they've announced a few of them, and there will be more. So I think that's going to be a focus, and so I think that y'all are raising some really good points. I would say that it's important to think about NSF as the entire foundation, because even though bio might not have a lot of investment in some of these spaces, other parts of NSF do, and we're partnering with them. And so these are really good. These are really good points.
Okay. Thank you.
Yeah, I was just going to, when you mentioned that, Paul, and also we talked about this kind of discovery. I mean, it kind of dates me, but I remember when I was in college that, we talked about, there was a lot of research in biomaterials around biodegradable plastics and what the outcomes of things like that were. And, things which look more sustainable and more, and I wonder where all of that has gone. I mean, single cell protein was a big thing in the UK at one point because of the oil crisis and all these kind of things that seem to have sort of faded away. And I wonder a bit about how much has been missed because we've slowed the basic discovery pipeline around things like this. They're all very bioeconomy related. And when you think about related to what you said and what Maria said, the burden aspect on the health, on the costs of public health as well. I mean, that's bioeconomy. It's not, you might not be making something big and shiny, but it's costing a huge amount of money to everybody.
Yeah,
Barbara.
I agree. And that's, go ahead, Barbara.
Sorry.
No. Okay. Yeah, and I think I'll just make a pitch for something that I don't work on at all and probably hard to understand, is agricultural systems, and food security is an obvious one. So again, an example, it was in the, using a plant as a chassis in order to get solutions sounds interesting to me. And it also sounds like a sitting duck for some pest or pathogen that's going to exploit that particular organism. It's like, do we really need another Irish potato famine, because of this? So sorry, that was a little snarky, but I think that this is where we have to be careful. So that sounds like an interesting solution on paper, and please, any botanist in the room, speak up. But it does worry me a little bit that, I don't know, for example, in the microbial world, I don't think people are assuming take E. coli and use it to solve all problems, and you can have a bacterium solve a problem. I mean, it's the whole idea is you look at other evolved systems and how they might solve the problem. So I found that particularly interesting, but again, I don't know anything about botany, and maybe it is a good idea.
Yeah, food system is another classic of, you know, a lot of people thinking about climate and food systems, and looking at the impacts that are direct, right? But not about things that we already know, like depending on how you plant, you know, there's so much more traditional knowledge, how you plant, where you plant a crop, what it's surrounded by has a big implication for who comes to eat what. You know, there's a lot more that could be run in that area, but that we can find, or is considered basically. You know.
But that's a very good bioeconomy strength. I mean, that would play to the US bioeconomy. That would help agriculture. It would help the consumer. It would help everybody. I mean, and like you said, I mean, it speaks to that the complex. I still think that this is where the AI and machine learning aspects come in. We talked about this a bit last time, and Karen Mongell was very receptive to this. They're using this to develop testable model systems where you actually do the experimentation and you look at how a system works and you question it. Because if you look at the challenges in healthcare and trying to re-engineer a human microbiome, that's still not easy, you know, to put it mildly. That speaks to so much work, and we still are so far away. I mean, just not understanding how a complex system fully works.
This makes me think of actually a couple of things that are already taking place. For example, a former colleague of mine at USC started a company. Well, actually, he took over from a family member company that was doing, and the only way that I can explain it is probiotics for the soil. Okay, where, you know, putting the right things in, and it all depends on, you know, where the place is, how much rain it gets, et cetera, et cetera. But these farmers who are getting this stuff to put on their soil were incredibly resilient when things changed. They didn't do much better initially in terms of crop yield, but when things suddenly changed, they were very resilient. Okay, so and General Mills, by the way, is very interested in this company. Not surprising. Okay, the other was my first graduate student started a company when she sold recently, and fortunately, he still seems to like me. But what he did, instead of looking for algae that might give you biofuels, he said, what crops are we already doing well? And there's one called Carinata. It's related somehow to canola, guys, but I'm not quite sure how. Anyway, he genetically engineered it to produce an oil that has been used in many jet planes already. He was in LA about 2 years ago talking about United Airlines flights that are 25% now with this biofuel. But instead of going to algae and how do you scale up, he said, what's already scaled up? I'll just engineer it to make what I need. And I think that it's approaches like that that are going to be much more successful. And again, I'm going back to what Maria said earlier, that, you know, you use what nature's already given you. And in this case, what agriculture gave you, but I think more attention on that, you know, how organisms are adapting, how we can use the present day situation for our advantage.
Yeah, and I want to talk a little bit about, there's another question about bottlenecks. And I really like the example that you brought up, Maria, because I think that brings up a reason why these approaches are not more common. And this one has to do with, you know, I don't know if you know this, but forest restoration projects the vast majority of them fail. It's kind of the present that is the truth. So what happens is that in many cases, because in Brazil, this is a case study that somebody was telling me about, people are reluctant to work, say we plant something or research something with natural species because the outcome is uncertain. And we plant eucalyptus and you let it grow for 50 years. And you know exactly how much carbon you're going to get, or you can make a prediction of how much carbon you're going to get. But what the natural system gives you, what the eucalyptus doesn't give you, is much more resilient. So the example that you gave with the soil probiotics, perhaps it doesn't have the same effect as planting that soil fertilizer using somebody in a crop. But it protects you under conditions of instability. And I think, companies generally don't invest in these approaches. We don't invest in these approaches commercially more directly because there's more uncertainty on the outcome. They're more complicated. And it's easier to say this simple solution, it works, we can predict what it's going to do until, of course, it doesn't. And so it's there's that caveat. I think commercially you have to assure the board members that whatever you're doing is going to be profitable. So the risks are the small companies that take risks and they test out the systems.
So we end up with two aspects of this that I think in terms of I think that's almost, I talked to someone about this. I think I was talking to Simon about it last night. This is almost not our problem, some of this, I think. If you look in Switzerland, they invest vast amounts in basic research. These people are not all doing biopharma research. The idea is you have a strong bioeconomy built on well-educated people who know how to do discovery-based science, who are rigorous, who are well-trained. So I think part of that leaks into workforce. I think it's about making sure that, to what you said, Maria, that people are incentivized to do basic research without having to think if they're going to go find, or if they're going to be entrepreneurial. Some of us just aren't entrepreneurial. It's just not in our mindset. I'm not that person. As an aside, when I worked briefly at the University of Copenhagen, the VP for research there had an idea how to make everybody entrepreneurial. It's that if you had an idea they subsequently found out should have been patented, they were going to dock your pay, which seemed to me the worst ever incentive scheme I'd ever heard about to make you entrepreneurial. It really was just. So I really think you have to separate these things. To what Paul said, the biofoundries, the incubators, you have to let people run with it if that's their thing. And some of them people are really good at it, and they just need an opportunity to do it in a way where they're doing it for themselves. They're not having to compromise, I think, with too many outside interests. They'll draw inspiration from it. I mean, I think it's kind of telling what Maria said, that the person who was successful took over someone's company in the family. It kind of gets you past the first hurdle. I mean, I'm not trying to be funny. I think it really speaks to that point about how you enable this. People have not got to feel there's a first hurdle.
I can't remember exactly what was said earlier about having people who were IP experts being embedded with, I don't know, with some of these programs, so that people who didn't realize and would have had their pay docked in Copenhagen, we're paying more attention to, or we're given advice about what could be patented, what might be important, even if the researchers themselves didn't do anything with it.
I can ask, don't NSF, they did have, or do they have a program to try and help PIs? transition ideas into through this? I'm trying to.
Yeah, I mean, that program is in TIP.
Yeah, so TIP is trying to do that.
Okay, great.
Interested.
Yeah.
Yeah, I mean, I think that there are programs that are now in TIP, used to be in engineering, but there are programs in TIP that really do want to try. The I-Corps program, for instance, was intended to try and help people learn how to become entrepreneurial and how to do that in a smart way. So yes, it's definitely something that NSF has supported that kind of program for a really long time.
Right.
So in the last 10 minutes of our brainstorming session, is there any big, okay, obviously I'm not going to, you're not going to say this is a public meeting, but is there any obvious big research idea? Obviously this is your secret research idea, you should not share it. This is being publicly broadcast, the thing that's going to let you buy that Caribbean island you've always dreamt of. But is there a big research area you think that NSF or just the US research program is missing out on? Something a little bit out there that is worth doing that just needs to kind of be kickstarted?
This isn't really answering your question, but some of the, what we appreciate in biology at NSF, many of us, is the evolved solutions on the planet, right? It has given rise to the biodiversity that we appreciate, but that's disappearing very quickly. So I find it ironic that there's a lot of interest in bioinspiration as biological species are declining. So maybe the point is to look in unusual places before it disappears. So it hasn't really been until recently parasites listed on the IUCN Red List, and still there. To me, arguably, that is the most common lifestyle on the planet. So until you start really listing things like that, you're not really appreciating the fact of what's disappearing. I live in Connecticut, not that far from Lyme, and I can't think of anybody in Connecticut who likes ticks, unless it's actually their research. But species diversity in ticks and blood anticoagulants, this is big stuff. And so I'm not trying to save the ticks. My point is I'm trying to say that if you look at weird life, you can find cool solutions. I think some of this came up earlier. This isn't a big thing. I think I'm actually saying an obvious thing, is that conservation biology doesn't really get a whole lot of respect now, which is quite sad. So if there's anything that bioinspiration can kind of give us a little bit of also inspiration to conserve species, even if it is for selfish reasons that we're trying to find solutions to human problems, you still might get the outcome of some better species preservation efforts. This is already on NSF's radar, though. So I don't know, Mike, that I'm really saying anything new here. It's more about appreciating all the weird and wonderful life on the planet as a possibility, even those that, I don't know, we tend to fear. Things like venomous snakes and ticks, and I don't know. Well, maybe one that we don't fear, but until recently, it has been way undervalued what endophytic fungi are doing for plant interactions. So this is like, you have to kind of break things down and understand their complexity in a way, a little reductionist, but to start to, so maybe I'm saying honor the symbioses, right? So this kind of weird life includes interactions between species that are just profoundly strange, but we might be able to be inspired by those for solving some problems. I think I'll stop rambling now because I'm not sure I'm making any sense.
No, I think so. This is I think it's a really interesting. It's kind of it's kind of to the side of what Teresa was talking about, because it's looking at things you might understand pretty well individually, but heck, you don't know how they work together or what they're doing in the same space.
What Maria, I keep saying Maria, but there's two Marias. I'm sorry. When Maria Uriarte was saying earlier about honoring and understanding the biotic and abiotic complexity and how it interacts and the whole sort of systems approach. I just gave you one quick example of a thing that is a system in a sense. It's a species interaction that has evolved to be one that is resilient to being broken apart. Right? I mean, it could be parasitism, it could be mutualism, but it's still something that persists. So that's kind of weird biology that we could tap into. Does that help at all, or is that more confusion?
Karen would like to share her screen if we permit her. I vote yes. Everyone said yes.
Just because you're. Remind you of this new partnership between NSF and the Paul G. Allen Family Foundation that focuses on conservation science and ecosystems. So I guess I'll go back and say that there's a lot of this kind of science supported at NSF. And so not everything that we're going to do is going to be biotechnology or bioeconomy related. So I think that I'm certain that you'll hear that message again this afternoon from Susan, but just want to let you know that because we're not talking about it at this particular meeting doesn't mean that it hasn't been, and it doesn't continue to be a focus of the basic sciences supported at NSF and Bio. Over.
Thank you for that reminder. Do we have anything to report back? I feel like we're just kind of.
I want to hear what
you have the notes, right? You have some notes, so maybe.
So, Barbara, would you like to summarize? Barbara's, I can see she's been writing a lot of stuff down because we've been.
Writing down all these examples, but
turn your mic on.
Your mic.
Oh. Okay. Yeah, I've been writing down all the various examples that you guys have been providing, but I don't think I have a good sense of, how to pull that together into a big opportunity, or, it's sort of missing stuff that we need to make use of. And so I don't know. I am the note taker, but I'm not reporting out. Am I? Who's reporting out?
Yeah, I'm reporting now. But, can you share the notes here with everyone, or do you have to go through email?
I've been doing it by hand, unfortunately. I'd have to transpose them. Ugh. Sorry, that was a big mistake, yeah.
Can you take like a picture of, a JPEG or a PDF and send it?
I'm not sure they're going to help you. Yeah, I was probably doing too much note-taking and not being involved enough in the discussion.
So I had some points. I mean, Barbara can send you some elaboration. So we talked about, I really liked Maria's headline. I think this should be a review paper. Nature-based solution, carbon world nature-based solutions. That was something you talked about. I kind of liked that. We kept on coming back to the better understanding complex biotic, abiotic systems and their interactions. How little we still understand them fully. I think we talked about primary and secondary benefits for the bioeconomy. Paul talked about difficult questions. I mentioned we don't know most of what a cell does still after all this time. And that's got to be a fuel for the bioeconomy, just finding out all of that. I don't know if this comes down to us or engineering. I think we're still short of it. We talked about this a bit in the last meeting about instrumentation and monitoring, how you get at some of these questions. But I don't know if that's us or not, Karen, the need for technology. Whether it's biologists telling people what they need from current technology and whether that links in. So that it's not only the big expensive equipment side of it for proteomics or other things, but I would think it's for complex model system building and analysis that we need.
I mean, were some keywords also the measurements and surveillance of what's happening?
Yes, yes, that's right.
Okay, the natural world.
Can you put, I mean, Michael, if you have them electronically, can you put them in the chat?
Yeah. These are very crude notes.
Yeah, we're working with whatever.
I can, let's see. Just going to offer to hand you my notes, Maria, but let me take pictures quickly and I'll send them. I think there's sound bites there, but you know.
We didn't get very much. I mean, I was wondering, it's almost like, I don't know if this is practical, but a colleague was telling me the other day, he's like, yeah, sometimes when I'm thinking about, these problems, I like to have somebody that is from engineering where they have a completely different and they come to, and I feel like some of these programs should have an ecologist attached to them. Okay, great, this is great, but like, do you really think this is going to work in the real world? And, just to sort of, a reality check.
I think we're on a brief break right now. Right?
Yeah, we are. I'm just going to type into.
I'll be back.
All right.
Oh, I'm just taking a picture.
All right,
so
I'm
she can
typing
read
into
them, right? What a
in
stupid
Maria.
thing to do. But I was trying to watch people as well. What is Maria's email? Do you guys have that? That was it. Jeepers. A couple of comments I took note of. Learn how to study.
Teach them how to learn. Good.
That's on.
No, that's really important. Right. Run to the bathroom first.
Just try to transpose it. There we go. Okay, she does have them.
What?
Yeah. Honestly, I really enjoyed that. Ohio. I think it was wide open if I was going back to Columbus, Ohio. Or Jim Jordan might meet me downstairs. So.
Maria, can you read my handwriting? I don't know if you're working from my notes or Mike's.
I am starting with Mike's.
Okay.
And then I'm gonna open your notes. Did you?
So sorry.
I don't see it in the chat. Did you send it by email or?
I sent it by email to
MU.
Yeah, MU2126.
Yeah, I'm just writing myself some notes. Let me check.
Okay, all right, okay. Sorry.
Let me check that you're right. No, it's fine. Let's see, because sometimes my email, yeah, I got them.
All right. Apologize that it's handwritten. That was so stupid.
No. I think, yeah, no, thank you. You took a lot of notes.
Yeah.
I'm gonna try to write some notes. I'm just, so I can be more coherent. Let me just remember a lot.
That's what it says.
Yeah.
Not gonna matter, I'm gonna be sitting all day.
I brought a sweater for the cold.
Well, I was just trying to see if I could actually.
Let me
Connect.
Glad I brought my pullover with me because in case, sitting around all day. Yeah.
All right, that's the breakout links.
Hi, how about you?
Okay.
How's being dad doing?
I'm much happier being dad.
Really? Interesting.
Cider, right?
That's
Very much happy about being dad again.
It's been good. Still getting into the new.
It's the limp.
You're right. River, she's really like the school.
A weirdo.
Vicky likes. So that's where it's been kind of interesting. It was like, other things that are super. I can't complain.
Did you have snow before you left? I see you wearing your boots.
Oh yeah, no, I just use my boots. I go. Many years ago, I dropped something very big and heavy on, and now since then, I've seen you too.
Yeah, I noticed you didn't even offer to answer a single question.
Well, I was a
Oh, I would have loved to. I would have loved to. No, not ours. Not at all.
It's all about manufacturing. Aren't you?
That's what I was all about.
My daughter's an AI.
Everything?
I mean, first of all, I was like, can I remove things? When do you stop?
When you stop with that, yeah,
That
exactly.
One serious video.
I just like losing the next one. It's like I don't want to be the top.
Pick your favorite medicine.
I'll pass on the financial independence thing. Exactly.
This is getting so.
So are we ready? I'm going to be purely selfish and go in reverse order because I can see that my reporter is still, our reporter is still writing because we made her job extremely difficult.
Michael, I'm happy to go. I'd rather go first because I
You just want to get it out of the way. Maria, is that what you're saying?
Yeah, pretty much. Is everybody here or do we need to?
It's going to be, Maria, it's going to be impossible to follow us. I mean, you know, what me, you, Paul, and Maria and Bob came up with, I mean, it's going to be, anyway. All right. So welcome back, everybody. I hope you weren't all warned about your behavior because you were online like group one was, but anyway. So we're now going to hear from each group. So does group one, the ones who stayed in this room, have someone who would like to present? Yes, her name is Maria Uriarte. Maria, would you like to present the outcomes of our discussion?
I would. We had a very philosophical and broad-ranging discussion about this topic. And, you know, several of us were concerned that the report generally had a very strong focus on sort of commercial approaches to the bioeconomy, where there's a very clear value to commercial enterprises underlined, but less focus on research on approaches that had preventative or beneficial impacts with a more sort of diffuse, but potentially broader impact on society. And these are approaches that build on what nature already has to offer. And I will offer 3 examples from what we discussed. The first one, Maria Pellegrini brought up an example from, I don't recall exactly if it was a former speaker or somebody else, from a small company that developed an approach for fertilization that relied on probiotics in soil as a way to protect farmers from fluctuations in crop yields under uncertain environmental conditions. And that is very different from, say, you know, an approach that focuses on developing a particular plant type to increase yields. There was a second example that she brought up from the use of oil from Acanola relative that is now being used successfully by United Airlines to fuel jets as a proportion of their fuel. And these are both approaches that—
Maria?
Yeah.
Sorry, Susan's having some trouble with clarity on your audio.
Oh, I'm sorry. Let me get my, I thought
It's not just Susan.
Well, Susan's next to me and she said it to me.
It's just, I don't want to miss.
Susan
Yeah.
to feel bad.
I don't feel bad. I just didn't want to miss what you were saying,
Yeah.
Maria. Thank you.
So can you hear me better now?
Yes.
Yes, thank you.
Yes, we can.
All right, so let me start over. I'll try to say it better.
She needs to change her microphone to that headset.
So what we discussed is that the report had a very strong focus on commercial.
Yeah, tell her.
Maria, sorry to interrupt you again. Could you change your check your microphone setting and make sure it's on your headset now? Because it still sounds like you're coming out of what your previous setting was. That you're.
How about now?
Oh, perfect.
Great.
So much better. So much better. Yes, thank you.
I'm glad we worked that out. I'll start over. The discussion that we had to do with the general focus of the reports that we read in preparation for the meeting. And we felt that when thinking about the economy, the bioeconomy, the reports generally focused on commercial approaches to the bioeconomy, where, generally manufacturing based and a clear value to commercial enterprises down the line, but much less on research that focused on approaches that had preventative impacts with a more diffuse but potentially broader benefits to society. And these are approaches that build on what nature already has to offer. And I will offer 3 examples that we discussed. The first one was Maria Pellegrini spoke about a company that is using probiotics for the soil as a way to protect farmers for fluctuations in crop yields that occur under uncertain environmental conditions. And this essentially is using a nature-based approach to protect farmers from crop losses. She also brought up an example of the use of a canola relative to develop oils that are now being used by United Airlines for fuel. The second one is for nature-based solutions for carbon capture. The reports primarily discussed, sort of carbon scrubbing or technology approaches to carbon capture as a way to mitigate carbon emissions, rather than nature-based solutions, which is something that has been broadly adopted by the corporate world, but has not been very carefully implemented and requires a much deeper and better understanding on how to approach and monitor what is happening with existing forests and regrowing forests. And again, it's an area that was not mentioned at all as part of the bioeconomy, even though it could have potentially a huge effect. And the third example that Paul brought up is this what is being discussed as the One Health approach to the study of human disease. We can develop drugs and apply them using precision medicine, but what are the processes that determine disease transmission? How can we leverage what we know about existing organisms to add to the bioeconomy? So essentially, these examples that we were discussing was essentially a warning about the seduction of simple manufactured solutions, but also to consider as part of the economy, the complexity of nature and what we can learn from it and we can use it. And we thought that this included things like advancing our understanding of biotic interactions in complex systems, generally a better understanding of fundamentals of biological systems, genes with unknown functions, symbiosis, parasites, and so on, and understanding climate change and its impacts across the bioeconomy with potential, again, preventative benefits. In terms of bottlenecks, we discussed technological bottlenecks, both as an absolute and local limitations, and the importance of developing a climate for innovation and entrepreneurship that does not distract from basic research. And I understand that some of these bottlenecks will be addressed in the tomorrow when we talk about infrastructure or later today. So that's what I have. So if people from the group want to add, what I may have not mentioned or forgotten.
Does anybody? I thought that summarized things well. Does anybody from Group 1 want to add anything else? right. Thank you very much, Maria, for working through our semi-cryptic mass of data we gathered. That was we were showing the data problem there in a microcosm. All right, so thank you very much. Breakout room 2, who will be reporting for breakout room 2?
So Kennedy and I will both be reporting. So I'll start off by talking about the opportunities that could be leveraged. One of the things that our group came up with was trying to identify the products. What are the products that we can imagine coming out of the bioeconomy? And of course, one of the big products that we saw as low-hanging fruit was the human investment, the human capital. Having people get trained early and often throughout their career so that they could contribute to the bioeconomy, either in obvious ways or less obvious ways, as they move through their educational process. So that's sort of ties in with part of what we identified as bottlenecks. Right now, oftentimes students are not getting introduced to what could be components of the bioeconomy until later in their education. And so trying to get them introduced to these things, basically in the K through 12 setting. So investing in the human capital at that point. Kennedy, I'll let you jump in.
So I'm just going to follow through with the bottleneck. So apart from that, we also talked about a topic that we had previously discussed. That was science in the U.S. in terms of postdocs and scientists, about how much time they are staying in their positions rather than having the specific positions to work as a full-time employee at one of these activities that have to do with the bioeconomy. The other bottleneck we talked about was about divided communities. So the idea being that the bioeconomy of one area might have in a community that is divided. So it kind of goes back to that the idea about when you bring in a community report, what part of the community is being vocal about the idea or the event that you are actually there to list to get ideas from, right? So the idea is that you might be in a small community, but it's still divided. Example that was given talk about simple thing as climate change, right? You can go in one community and half of them believe in climate change, the other ones do not believe in climate change. So whoever is more vocal is what you'll eventually get about it. And that's the same thing that would have in terms of bioeconomies. Then the other thing we talked about is skill set. So the idea was about training people to work in the bioeconomy in terms of how we can change the way we train and provide skill sets rather than this, terminologies that we give people, right? So the idea is that someone might have great computer science skill sets, but they don't want to be a computer scientist, or they don't know what to do with their skill set. So the idea about the bioeconomy is if we train the workforce in specific skill sets rather than to a specific job, then they'll be able to use that skill set in any situation rather than a specific situation when you're talking about the bioeconomy. And I think that's all I had. Did I forget something?
Thank you.
Thank you.
Okay, so that's really good with very different directions the first two groups went in. So can breakout room 3, who's going to be reporting?
I'll be reporting on behalf of our group, which was Kimberly Albuquerque, Gretchen Norch, Tom McClung, Eric Grotwall, and Tom Daniel. So we actually started off with talking about a paper where people are using Escherichia coli to do electron transfer from wastewater. And, I was thinking it's like two birds with one stone, that we're solving two problems with one kind of solution. Didn't really elaborate. I don't really understand just how much energy is being generated, but, any with an approach like that seems like worthwhile. Then we got into a conversation that Tom Daniel led, and I'm gonna let him describe it. Something to the effect that living systems and proteins compute much more efficiently than anything human generated. And then he talked about these compute farms. So Tom, you want to elaborate on that a little?
Not a lot. Definitely.
Hank, can you log into Zoom?
I am in.
You in?
Am I not there? I can see everyone.
Hank, I think you captured the sufficient detail. I'll just leave it at that. There's lots going on in how natural systems process information with efficiency levels 10 to the 4 greater than synthetic. And the part we were talking about is as compute becomes ever more the energy hog and water hog, as it turns out, our attention as NSF should turn to more efficient compute platforms that are inspired by or actually driven by natural systems. I'll leave it at that.
Great. Can y'all hear me all right?
Yeah, we can't see you.
Just can't see me. Thought I was, I mean, I just signed in.
Is your video on?
Maybe not. Not even seeing my controls here.
Hank, if you signed in with the link that's at the bottom of the Word document, that takes you to a webinar and you need to be elevated as a panelist.
Oh, you're right.
is he an
I'm in a
attendee?
webinar. Let me get to the right link. Or do you want to just keep going?
I think because it's being.
Oh, for the people, yeah.
Oh, yeah, he's an attendee.
Where's that link?
Instead of a panelist.
I went to the.
He's our
webinar,
only
but they
attendee at
wouldn't.
the moment.
Hold
So
on.
I feel loved.
That's the SharePoint.
Let me send a note.
You sent to me?
No, I think.
Why don't you log out of where you are now,
Okay.
and then we'll get you a new Zoom link to check.
No, actually just stay there.
Okay.
Stay there.
You're going to transfer.
Let me see if I can get.
Catch me in.
Okay, so I'm just going to jump in while Hank is out there and talk about what I remember most. And that is Tom came up with the term stackable certificate. And so we were talking about one of the bottlenecks and one of the reasons that students may not be so excited about the STEM field is it takes so long. It takes so long to get from point A to being postdoc and then practicing scientist. So maybe we could shorten that. But if we shorten that, then basically they get the skills they need and they're excited, and then they go to work, but then they can come back and learn some new things by getting a stackable certificate. And so the stackable certificate is something that you can get as a scientist in industry, and you come back and you get another one in academics. And then in the meantime, you're there at whatever institution, and you can teach your industrial skills to the students. So it's a two-way exchange, and the students could get a stackable certificate too. And what that means is you get a certain amount of expertise quickly in a module. And so basically you have general training, but you need specific training to solve the problem that's facing you right now. So you could get just something that would say, okay, this person is ready to do something good with the carbon that is captured.
Yeah.
Excellent. Thank you for jumping in, Gretchen. That was perfect timing.
I just wanted to see if we could focus that a bit to actionable items for us as Bio NSF, because obviously we can't decide what constitutes or comprises a PhD at an individual university. So when you were discussing it, were you discussing I mean, in my head, I'm already thinking about various ways, but something to promote getting to that kind of thing, because it does seem that the big goal is outside of our purview, but we can incentivize maybe. So did you discuss that in any way of how we could?
So I think we did kind of touch on it a little bit. We have to change the way we're preparing students. And it did mean, changing it so that it didn't take as long as Gretchen mentioned, but also that we're teaching them to learn not only the technology of today, but the technology of tomorrow, right, of 10 years from now, when everything's going to be that much better. And it's not, in the way that we're educating students today, we're not really doing that.
So is that a bottleneck then?
That's a bottleneck. It's a bottleneck related thing. Yeah.
And we did not discuss it, but I think an opportunity that's also there is how we think about broader impacts and what the broader impacts really could mean when it comes to providing some sort of a degrees that are more opportunity driven or short-term driven, like a certificate.
Yeah, that's where the whole idea of the stackable statistic was mentioned, where people get this, a relatively short-term education program, but it results in a certificate that assures that you have a certain capacity to do some work. And this was another one of Tom's ideas, that you could stack as many certificates as you need to have competency in this new area of research.
Could I just add a little nuance to that, if possible? Yeah, so these are existing programs in some institutions around the U.S. There's another feature of them is they're very accessible. They're not huge courses in time sinks. So biologists can get a certificate in data science or, et cetera, et cetera. The other side of the story is industry has an interest in this as well. So their practitioners, their scientists or researchers or whatever also can get stackable certificates at institutions. NSF could actually have programs that would help support these partnerships with academia and industry. So to an actionable item is think about that as something towards the bioeconomy.
Yeah, no, that's what I was thinking, because I too know about the different universities. My own bottleneck in thinking about this is when it comes down to practice, most people are thinking about how to harness such a certificate into a money-making venture for their department or college, because and so that sort of complicates and gets in the way. And so in my head, I'm thinking, well, how can the NSF be more purpose-driven than money-driven for this? But thank you for bringing it up.
Great point.
I think also one of the things we were thinking about was, and Hank addressed this briefly, but the importance of switching into a mindset of ongoing continuing education, because as Eric said, he has no idea what we'll be doing in 10 years. And so the skill set that we develop today with these certificates won't be applicable in 10 years, but what we need is an ongoing process, say akin to continuing medical education, where it just becomes normal that you acquire new certificates for whatever is relevant as the economy changes.
That's all I have. Does anyone else in the group have anything to add?
Can I pose a
which was, we're sort of resonating with a lot of this either synthetic cell biology or de novo protein design. These are all emerging opportunities that I think have incredible potential for accelerating new materials, new devices, new sensors and things like that. So I just wanted to, that did come up.
Yeah, they did. Thank you.
Just something for you guys to think about, maybe to give some advice is, I think you're getting there in that the mentality of trainees right now isn't really realizing that whatever they do in graduate school is actually statistically unlikely to be what they do later on in life. And that seems to have lost track to many people, that it's about learning how to think, learning how to do things. So I don't know how to bring that back, and that might shorten also what the PhD is and what it isn't. But It's just something I'm thinking about, but I don't have any answers.
Anybody got any questions for any of the breakout room report outs or anything else you'd like to add? Yes, Maria.
Sort of a sub-theme that came from our nature-based solutions idea had to do with funding discovery science and not just hypothesis-driven science. Just wanted to mention.
Okay. If we have nothing else to add, so I put something in the chat. If you could all send me from group breakout rooms 2 and 3 your notes, because this will be one of the themes that we want to discuss with the director. If anybody feels passionate and eloquent and would like to be this person without me tapping you on the shoulder, email me. Otherwise, someone will get tapped on the shoulder. Just to give you a heads up. All right, I don't know if Shree's online yet, but Shree's going to give us a presentation at 3:30, which will actually, again, kind of dovetail into some of the stuff that we started to talk about, some of the infrastructures that we need. And I think this is a good juncture at which to sort of ask questions about how this will enable some of the things we talked about in our breakout sessions. So Shree, do you want to wait a few minutes just in case anyone else comes on who's seen the agenda? They might want to wait until 3:30 as well.
Absolutely.
All right, thanks.
Just to save time, I'll just put up my screen anyway.
All right, so we're back. So the bold goals report identified a number of areas where new or additional investments in infrastructure are needed. NSF supports infrastructure through several programs, and BIO has contributed to a number of these. To introduce this session, we will hear from Sri Rahavachari, a program officer in the Division for Biological Infrastructure. Sri?
All right, thank you all. Some of you might actually recognize me from my time in iOS, particularly Scott and Suzanne. But thanks for giving me the opportunity to talk a little bit about the infrastructure programs at NSF and how BIO is taking part in this. And this has come up today on several discussions and breakouts that advances both in basic discovery science, either hypothesis-based or discovery-based, as well as sort of applied biological sciences, really is underpinned by instrumentation and large-scale infrastructure. And these are exciting times because both advances in physical sciences, as well as all our tools in biological sciences and the data revolution and AI, really make possible new developments in large-scale infrastructure and really providing access to all of these infrastructure. And this is much more imperative if we want to make progress both in basic research as well as translational aspects. So, next slide. Again, NSF has several programs that BIO participates in extensively at multiple scales. So we have at the low end, the Major Research Instrumentation Program, the Mid-Scale Research Infrastructure Program, which is sort of at a slightly larger scale. Then there's the second Mid-Scale Research Infrastructure Program, and then Major Instrumentation Facilities, as well as Center Scale Infrastructure. And I will talk a little bit about each one of these and how they touch upon both the bioeconomy and fundamental discovery-based science in biological sciences. Now, this is the same slide that Teresa showed a little bit earlier, and I'm showing it again just to underscore the fact that all the investments that NSF and BIO are making in the bioeconomy are really well aligned with the R&D needs that are outlined in this report. And just for reference, I'll use some of these icons as I introduce different aspects of infrastructure programs that we have and how they might contribute to these goals. So again, many of you are probably familiar with our Major Research Instrumentation Program, which is sort of at the campus-based infrastructure or individual PI-based infrastructure or instrumentation. It could be instrumentation, cyber infrastructure, databases, compute, etc., which all are there to expand our capabilities to build and measure biological systems. And we have two tracks in these. One is both sort of new design of instrumentation, as well as purchase and installation of instruments that are more widely used around the campus. And again, an important part of the MRI program is to integrate research with education so that academic departments really can integrate across different disciplinary units and also have regional collaborations to really have well-equipped research environments that are important for discovery-based science. In the last few years, Congress recognized and NSF also stressed heavily that there was a big gap between the Major Research Instrumentation Program, which ended at about 6 million in circa 2019, and then the Major Facilities Program, which started at $70 million facilities and instruments. And to fill this gap, NSF started a program called the Mid-Scale Infrastructure Program, which some of you may be aware of, but perhaps not many of you are. So I just wanted to give you a brief introduction to what these programs are. Right? So again, these programs are aimed to fill the gap between these two limits of what can be acquired in terms of infrastructure. So the Mid-Scale R1 program supports both the implementation and design projects for infrastructure in the range of $4 million to $20 million. That's the current limits for these. So the idea is that a shovel-ready project can essentially be brought in for implementation, but if groups or teams have ideas for design a piece of infrastructure that again fits in this range, then they can propose a design project as part of the Mid-Scale R1. In contrast, the mid-scale R2 program supports implementation-only projects. So these are projects that are in a very high state of technical readiness. They can be pretty much ready to go for implementation. And these are projects that range from $20 million to about $100 million. And important point to note is that these are funded through the MREFC appropriations. So they go through both an extra level of scrutiny, and the entire timescale for these are between about 2 years from when the letter of intent is received at NSF to when the awards are made. So groups that are interested in these really have to have a long planning horizon for actually putting one of these R2 projects. Okay, so the fundamental idea between the R2 or the mid-scale research projects is to really fill community-based defined needs, or essentially enable national research priorities. So in that sense, these research infrastructure programs really dovetail well with the bioeconomy needs. So as we outline what the infrastructure needs are, these programs exist to fill those gaps and needs, right? And the idea is that they will enable US researchers to really remain competitive in a global research environment. So in general, one way to sort of capture or illustrate the need is to perhaps go back and look at either the bioeconomy report in this case, or perhaps National Academy surveys or reports that really outline a vision for what infrastructure is needed. And in not only are they important for implementing this infrastructure, but then we would also have a trained workforce in the design and implementation of the science and engineering infrastructure. And that I will point that out in a second as I illustrate some of the projects that have been funded. So one of the projects that, again, was touched upon a little while ago at one of the groups that I was sort of listening to briefly and came up in the reports was a project from the University of Idaho, which is called the Deep Soil Ecotron, which really looks at both studies deep soil microbial and abiotic and biotic and abiotic factors, and even manipulate them and look at their impact, right? So this is a project that was funded the last round of the mid-scale R1 program, and is being implemented, probably be completed in 2 to 3 years. And then the other big success we've had with this, the bio has had with this NSF program, is that finally, and the US actually has now potentially several high field NMR spectrometers that are available to the US community, right? I mean, they're not actually installed yet, but these awards have been made, and they will be as soon as Brooker actually finishes them. But now we went from having essentially no high field spectrometers to having 3 of these in a way that is actually accessible for pretty much entire communities, right? Another one is, the most recent one is the Compact X-ray Free Electron Laser, which has been funded awarded to Arizona State. And this essentially takes a large-scale synchrotron-sized facilities, which are the only X-ray sources at this point in time, and shrinks it down to a 10-meter-sized accelerator, which can be essentially the design can be now ported, once successful, can be ported to pretty much any campus. So in that sense, all of these projects really advance our capabilities, in some cases for structural biology or more environmental biology. And they also train students, postdocs, other trainees and scientists in how to go about actually implementing these large-scale infrastructure projects, right? So it's not just learning to use them, but learning to design and build these pieces of infrastructure. At the other end of the spectrum, we have sort of intermediate or center scale research infrastructure, which NSF and Bio has been supporting for several years now. So some of these, again, include structural biology field, so BioXFEL, which is another X-ray free electron laser facility, the Cornell High Energy Synchrotron, which provides beamlines for structural biology, as well as the National Magnetic High Magnetic Field Lab. All of these are centers that NSF supports for physical instrumentation. Similarly, we have several projects or centers that we support for the cyber infrastructure piece of it. Probably the most famous is the Protein Data Bank and Cybers, as well as iTech Bio, which sort of provides integrated digitized bio collections for the large-scale community. Okay, so at the other end of the spectrum are the really large-scale multi-user research facilities. Now there are several of these NSF-wide. Of course, LIGO and the Large Synoptic Sky Survey, the Vera Rubin Observatory being some of the more famous ones. But for bio, we have the National Ecological Observatory Network as the only major research facility that we have. And these are, as you probably know, have 81 field sites across different regions in the continental United States and Hawaii, Alaska, and Puerto Rico, which sample and provide data products and really provide a large-scale data source for monitoring various aspects of environmental biology. Okay, so this was brought up repeatedly today about how do we harness the data science end of things and apply it to biology in general, and specifically perhaps for the bioeconomy, enhancing our ability for predictive modeling and engineering design. And the AI Institute program really fills one of those gaps. So this is a program led out of SAIS, the Computer Information Science and Engineering Directorate, which is a multi-year, multi-sector initiative which involves several partners, both within the federal government as well as the corporate partners, to really advance research in AI, both foundational and use-inspired, and accelerate the development of AI-powered innovation. So relevant here are several, for the bioeconomy, are several AI institutes that are now up and running. This is the third round of the competition, which largely are NSF and USDA jointly funded institutes, which are in the agriculture space. And specifically for bio, we just funded the Artificial and Neural Intelligence Institute out of Columbia University, which bio supported in part. And so all of these are examples of large-scale sort of AI ecosystems that the bioeconomy community and the biological community can really take advantage of. And important in the mission of the AI institutes is that they actually have to serve as nexus points for the community, whether it be academic research scientists or industry scientists, to really take advantage of these AI-based innovations. Right? So in that sense, this is really bringing the power of AI to several aspects, whether it's basic science or applied science. And I should mention, this was brought up again, is the notion of compute. And again, bio is really not involved right now, but we have been in discussions. It's led out of SAIS, which is a program called Cloud Bank, which really supports access to large-scale computing for various applications to a wide variety of NSF-supported researchers, right? And part of the AI Institute network mission is to also support access to compute. Okay, next slide. So one point that was made a little while ago was that all of these bioeconomy and synthetic biology really needs to have broad access. So this was brought up that perhaps not everybody has either access or ability to take advantage of either industrial scale processing or turn and test their ideas and their designs in the bioeconomy and biotechnology space. And so there's been sort of a need for a biofoundry. And what is a biofoundry, you might ask? Well, the way we've defined biofoundry, and it's been defined otherwise, is an integrated facility that takes advantage of discoveries in biological and engineered biological systems, and then provides access to instrumentation, workflows, tools, and processes so that we can actually implement these design, build, test, and learn cycles. And so the goal of these is to essentially accelerate biodesign, build capacity for innovation, democratize access to tools, and develop the next generation workforce, right? So all of these points have been made over the discussion just before this session. And the biofoundries, the need for biofoundries has sort of been outlined in several reports. So there is one sort of comprehensive report from the Schmidt Futures Foundation, which essentially says the US bioeconomy and charting a course for a competitive future. It is a little bit more restricted to the synthetic biology space, and one would want a more expansive definition of what a biofoundry would look like. But then there were several other reports in the recent intervening years, including the Executive Order for Biotechnology, which have broadly spelled out the requirements for biofoundries. And so to fill this gap, we just started this program, which is led by BIO, which is the Biofoundries program. And the solicitation was just released earlier this year. The letters of intent have come in, and the full proposals are due in about a month from now, a little bit less. And the idea is to build on all of the existing NSF investments to really take advantage of the advances in biology, in chemical biology, biotechnology, bioengineering, as well as AI and other innovations, to provide for an integrated facility where scientists, both in-house research groups as well as the larger community, can both engage in innovative research, build tools and technologies, and then provide broad access to the community. So essentially, anybody should be able to use these tools and technologies and instrumentation to go ahead with their, sort of build on their research. And eventually, these should provide a pipeline for translation as well. Right? So these are important for our innovations in biomanufacturing and also scaling up for process control. So that sort of gives a snapshot of what NSF Bio has really been involved in terms of the infrastructure space. So if you have any questions, I'm here to address them.
All right, thank you so much, Sri. That was very, very, very helpful. We had been talking about Biofoundry, so that was very, very timely. So I think it would be good now we have quite a lot of time to discuss this, because I think this is something that we'll almost certainly be wanting to bring up with Punt, the director, when we meet with him. So the kind of questions that we want to think about as a group are what priorities we should have in infrastructure, and we can ask Sri about how that's going and where we think, if anybody thinks there are gaps in what he just described and that might be important for the bioeconomy. And maybe a little more about how these facilities are accessible and usable to the larger. I think that was an interesting point that was raised. So I'll open it up for questions. Paul.
Yeah, it was exactly this issue of who has access to the biofoundries and whether it could be especially accessible to our colleagues who are least prone to be able to have these at hand. And you know what I'm saying. So can you say anything more about that? How ambitious could we be for?
Okay, so I can address that. So one of the goals of the one of the review criteria and one of the requirements of the Biofoundry solicitation is that 50% of all NSF support should be directed towards user-facing activities. And the biofoundries, and these will be all cooperative agreements, and NSF will have a heavy role in sort of the management of these foundries, is to ensure access to not just R1 institutions, but R2, PUIs, all kinds of institutions. And of course, well, the small business is a separate issue, but certainly make sure that everybody has access to tools and technologies at either essentially no cost or minimal cost, essentially for reagents and that such, right? So the goal, I mean, what has been integral to the biofoundries concept is really to democratize access.
Okay, that's great. Thank you.
Tom?
Thanks for a really nice presentation. You mentioned, and it was in passing, that there was some National Academy studies done on needed infrastructure. Can you, if I heard correctly, can you elaborate? Was there something specific, particularly relevant to biology or biodirectorate that you heard about?
Sure. So they're sort of balkanized. So there is certainly the report on NMR, right? So that's both chemistry as well as biology. And one of their recommendations was that the country, US as a whole, invests in high field spectrometers, which really provides access to structural biology that's not available right now. And essentially, as of now, I think Europe has an installed base of about 8 of these spectrometers, and US has 1 that is not available to the general public, general scientific community. It's a private instrument. So with this program, we've sort of filled that gap. So that's one of the reports that's available. There's a new one on physics of living systems, which sort of touches upon biology, but not really biology squarely. So there are examples. I think, Teresa, you may have ideas about other examples of National Academy reports that are more relevant to biology.
Kennedy?
I just want to follow up to the question from Paul. So you talked about access. What about in terms of regional? So part of access is based on also about the region where the founder is located. So how about the regional placement of the founders?
Well, those will all be considerations once we actually receive the proposals. We only have the letters of intent. And we will be, certainly that will be a big aspect of how we review these and how we sort of make decisions going forward.
Yes, good question.
Teresa, did you want to say something?
I popped it in the chat about the National Academy's report on safeguarding the bioeconomy that I think came out in 21. But I'm looking at Karen because I think we heard both of the same report outs that preceded the work that we did on the priorities of the R&D needs.
Susan's going to say something before we go back to you, Hank.
Sorry, I wondered, Kennedy, if you had something in mind, because the idea of the regional, there's a lot of competing things in your head about how to best, or in my head, how to best do this when you have very large scale, sometimes expensive things, and do you put them where the expertise is, or do you put them and hope the expertise goes there, et cetera? So did you have something in mind?
No, actually, you say the right thing, right? It's the idea that you have the biofoundries where the expertise are, or where people, because you talked about access from smaller institutions, right? So if you put up all of the West Coast, small institutions from the South, that's not access, right? So that's what I meant by in terms of regional.
No, I just, it's a complicated thing, so.
It is.
Yeah,
How to make sure they're.
Hank's hand is up, but I wanted to jump in on this. So by regional, I'm thinking there are some things that people just want to have access to for manufacturing that they can't afford. But I think that's different than what you're talking about, where there's a proposal put forward and it may be supported. I guess I'm a little bit more to the create it and folks will use it, but it's got to be flexible. So that wouldn't be useful for all users. It would be, I just feel like there's a lot of biomanufacturing that could happen and people just can't afford to do it with their facilities locally. And if there were several of them around the country, then there would be enough of this infrastructure that people could get access to it. But I think that's a slightly different beast.
Yeah, I hear you, Paul. I'm just take the example of high field NMR. There's not a lot of point in spending a lot of money to putting a high field NMR where there's not an NMR spectroscopist. So those are the things that need to be weighed. I'm not saying I know any answers. I thought maybe he did.
Hank.
Yeah, very, very good presentation. So my question is about the slide when you're talking about the centers, in particular iDig Bio and Cyverse, how those centers would work with the other centers. Are they going to play a synthesis role in this endeavor, or is it going to be somehow they're going to be federated so they could support the biotechnology enterprise?
That their design model and their access model is flexible enough that however the community wishes to avail of them, I think they're flexible enough to accommodate all of that.
Yeah, so if I could just elaborate a little bit. So, for example, iDig Bio is managing all this data that's been gathered from research collections, biological research collections. And it kind of gets to this, what can we discover about biodiversity? Cyverse is doing the same thing, but I'm just trying to connect that to what we're doing with biotechnology. Are we trying to get like new drugs out of the insects and things that are coming from these collections? Are we trying to use the data that's in those centers to synthesize something about, how we could benefit more the biotechnology? I'm just trying to understand that, because this I think it's really important, but I'm just trying to understand how it fits in.
I think there's going to be lots of different possible applications. It could be that as we mine some of the data that Cyverse is, allows us to access, it'll enable us to figure out what are the right sets of organisms that will enable us to degrade plastics, or as we compare, things in iDig Bio and layer on genomics data, stuff like that, that we may discover, there's some useful iridescent property that we could use in the next kind of organic photoreceptor that we would, or kind of a solar panel, and I don't know what those applications will be, but I think that we will never be able to imagine the applications if we can't mine the data. And I think that is, you've all been talking about the importance of discovery, and I think we all recognize that, but it is, but if we don't know what's there, we won't be able to be creative and imagine what you might discover and use it for.
Yeah, I guess the only other thing I'll add to that, and I see the importance of it, but I think that this effort's going to have to involve the people that are generating that data or curating the data. There are things that have to be verified and corrected. And I know a lot more about the iDig Bio thing because I've worked with that organization. But I'm just saying that you're going to have to engage not only the people doing whatever kind of technology that's the aim of the work of these centers, but you're going to have to engage the people in the museums that are generating the data.
Well, and I think as we think about the data infrastructure for the bioeconomy, that standard for data and metadata and all of that, and I am not the expert on that, I would have to defer to my colleagues around the room, but it's not simple. It's not just, it's there and let's compare it, right? I think point taken, and other people could talk about it with more authority.
Yeah, you actually identified sort of an important gap, because these are all at different levels and with different considerations of provenance and validity and vouchering and all sorts of things that all need to be sort of integrated. So so you've highlighted some interesting and important points that could form basis of what we need to think about, and you can probably discuss.
I had a question, and maybe this is probably my ignorance of the synchrotron in Arizona. So with those kind of new state-of-the-art instrumentation setups, did NSF, so I've had multiple parts of my question, so did NSF invest in the development of that, or just in the construction and installation of that kind of equipment?
Okay, so that's an interesting question. So that project has a very long history with DBI. So it originally came in, sort of went through the entire stage pipeline of infrastructure. So it came in originally in the MRI program to really sort of explore the first design. Then it came in and successfully competed for a Mid-Scale Research 1 award to set up the first sort of the front-end laser and the design itself. And then now they have a Mid-Scale 2 award to actually implement the entire accelerator itself, right? So it's sort of an interesting idea, because it's based on it's a principle for generating soft X-rays, which is somewhat experimental, but if it works, it'll really advance structural biology and several other fields. Right? So it's sort of very innovative design.
So I can add to that because I think they also had an STC, which you brought up, which was to think about how to use its XFEL. And so there was a structural biology community thinking about the questions they could ask to do this. I only know because I was just there yesterday. And I was really impressed by the success of this stage-wise, like the idea actually came to fruition. You get the little one, then you get the big one. And so the investment and the de-risking of the project, yeah, it may not work, but it looks like it's going to work. The other thing that was amazing when I was there is the amount, they have undergrads that are checking all the way from building the building. Is the rebar, each piece of rebar that went in, was it okay? And so that was some engineering undergrads who did that. So yeah, I walked into the control room and I thought, oh, I'm feeling older and older because these people, but they were undergrads in the control room. So it's that, I don't know if that answers your question, but they were on the whole path.
It does answer my question. The reason I asked my question is that I think that part of the discovery, but this is the kind of standoff almost between the discovery base and the bioeconomy around instrumentation, is that what drives the discovery is that you have an instrument that you can gather data that you don't know what it means, and so you gather the data and you figure out what it means, or do you know that you want a certain kind of data that you don't know how to gather, and that you then invest in exploratory instrumentation research so that you can end up with something to find that data that you couldn't find before. That's, what drives this? Is this economic need? Is it the discovery-based science? It's kind of a big question around this about which direction we go in. Sorry, I thought,
No, but you bring up an important point, and I think we've been flexible in bio especially, in encouraging all stages and recognizing very early on that we have to support pretty much every aspect of this pipeline, either from supporting conceptual ideas and then nurturing them through the implementation itself. So we have a standing Dear Colleague letter for either workshops or planning grants or design grants for these sorts of infrastructure. And again, and we've been very, especially the mid-scale program has been very broad with its definition of infrastructure. So since it's an NSF-wide program and we have our colleagues in social behavior and economic sciences, as well as the education directorate, community data sets are infrastructure under this definition. So just by the act of actually collecting that community data set and making sure that it is accessible to the community counts, right? So in that sense, we can really be very broad, and it's really up to the imagination of the community, whichever community is, whichever subpart of the community is, to really imagine what it is that they need, whether it's in the biotechnology space or the basic biology space, and then really propose it to us and have a really long-term horizon for getting these, right? So yeah, your point's really important.
Can I ask, so some people are interested in building it because it's going to stretch the time resolution. Now they're going to be able to see events that are very short in time. And I don't know what biological events happen in that short time. So, but if you build it, usually you find it. So there must be some, in my mind, compromise between just building this because if we have the capability, we'll find out really interesting things versus we need to address this question and so build that instrument for it. And I kind of think we need both, but yeah, I don't know.
Perhaps we could talk about all of the other ways that DBI, or Jim, the ways that DBI invests in kind of infrastructure development, whether it is through the innovation track or the ways that we do that kind of, we don't know what, or we think that there's a need for this and I'm going to develop a tool. So I don't know if that's Jim or Sri, but that might be part of the answer to this question.
I mean, I can just speak to the reorganization. Some of you were on the COV for DBI last time where you saw the reorganization of our programs around the life cycle of infrastructure, which is informatics, instrumentation, different types of infrastructure. So we have the innovation phase, proof of concept, high risk, something hits, we'll put it through capacity programs, scale it out. And, and Sri And Shri is our primary rep for the mid-scale program. So he's got his eye on the pulse of what's coming through those portfolios, and he fields all the calls from the investigators, and he gets his colleagues involved. Sometimes he gets us involved on these calls to help PIs figure out, which program's most appropriate for what they think is there. And, we only talk about the awards. We can't talk about the declines, but we're getting proposals, we're getting activity. Some fields are doing better than others. So it's something I think a lot of us on the screen are thinking about a lot. At least we've got programs now in that space with mid-scale that our DBI programs can feed into for something that might have that kind of capacity in the long run. I will say that, Michael, to your point about not knowing, how much definition do you have to have to your question to build that instrumentation, right? I mean, think of NEON. It's the other end of the spectrum. We're going to collect all these 180 data products at a continental scale, and we're going to let the community figure out what questions they answer with it. We knew what areas of science we would propagate, but it's I like what Susan said. It's like we sort of need a mix in there with biology, because biology is so all over the map, literally. And I don't know, Teresa, that's sort of, I do feel like we've been thinking about this. I've been thinking about it for a decade, and we've organized our programs to help the community come in where they're ready for project, even planning grants. We had planning grants in DBI before they went to the PAPG just for this purpose, so that people could get workshops and work together with their community to plan, what that next phase was going to be for that infrastructure need for their community. And as Shri says, very broadly defined infrastructure. So I don't know if that helps, but.
No, that does help. I mean, I think it's just very reassuring, to be honest, that this is not just that this is a key part of it. That's why I was asking. Does anybody else have any questions about this? Yes, Gretchen.
So I just have a question about the technical support once the thing is built. Is particularly, I guess, at the mid-level, it strikes me as a really dicey proposition. What kind of scientists do you have on hand to run it and maintain it, and or what kind of maintenance and technical supply does the investigator have to come up with? Is there a typical model?
Yeah, so there are two aspects to it. The NMR spectrometers are a different class by themselves. They usually go into an established facility where, and it's not just high field, they have a mix of everything, all the way from 400 megahertz up to now a couple gigahertz or more. So they have they have scientists and staff scientists who actually help the users, right? And the model for these is that anybody anywhere in the country can just send their samples and essentially log into a portal and access their results, right? So somebody else is going to run them. Okay, so that's the easiest part, right? So it's a little turnkey, push your button and you get your results. The other one, like the XFEL, is a lot more complicated, right? So you have to know what you're doing. But typically when we make these awards, we have the PIs put down a fairly complicated or comprehensive plan for operations and maintenance, which includes like who's the staff who's going to run this, how the user community going to be engaged, and how will a user actually, what are the user experience at their end? So some of these, like the XFEL itself, is very technical. So it's not going to be everybody, but then again, not everybody in biology is looking at attosecond reactions, right? So that's the space. But rest assured that we are making sure that people who need it will have the expertise to help them through. Now, the other level is the biofoundries, where we've explicitly built that into the solicitation. So they are required to hire a user facilities coordinator plus additional staff whose job is to help the users on their experiments or on their products or whatever it is that they're working with. So, I mean, in shorthand, think of the Genelia Farms research model in some ways. You can use the advanced imaging facility or some other of their facilities, and essentially somebody comes in, proposes an experiment, but there's technical stuff. So that's sort of the model that the BioFoundries is working on. Similarly, we have our colleagues in materials research who've put up the materials innovation platform. They also work on the same model. So we, at least through continued sort of management and stewardship through the cooperative agreement, NSF would sort of stay engaged in making sure that the user access and user experience is fairly smooth and seamless.
I'm going to ask another question which shows my ignorance and actually reveals a problem I'm facing myself. So, is there a more, I don't know if this is in place already. So when you put something in like this, and it comes a bit to what Kennedy said about the facility being far away, maybe you don't have on someone at your home institution who has familiarity with this technique. Do you think the NSF subsidize trainings or workshops to familiarize people with new technology and how to use it? Because they can't use the Janelia Farms model if they're not really aware of what it can do and whether it's applicable to their systems. So I'm wondering if there's a kind of training component that could be phased in. And I don't know if that comes through you or through someone else's office.
Well, it's actually incumbent on the BioFoundries or the infrastructure. So we did this with what's called the NeuroNext program, which by DBI and iOS jointly set up. And these were 9 large-scale sort of center-like investments. Again, they're not terribly large-scale. But one of the ideas, this is for neurotechnology. So think in terms of electrodes, viral vectors for labeling things, etc., or new optogenetic reagents. But then if you as a researcher in a small institution, say out, I don't know, you somewhere in a PUI or an HBCU needed to use it, you could avail of, so one of the things that these NeuroNext centers had to run was training courses and workshops. So they engaged MBL, they engaged Cold Spring Harbor heavily to train people in using these, and then sort of what was possible. And then people would go there to one of these centers and actually access these, right? So we've had people sort of do really well with these. And that's sort of the experience that we're sort of leveraging at this point for the BioFoundries.
All right, thanks. Jim.
Yeah, I've been wanting to say this, and this is maybe the time to say it, which is talk a lot about access and open access, democratizing the science. It doesn't happen with training. It just doesn't. These things, the communities aren't that big, in a lot of cases to democratize in the way we all aspire to democratize. So, for example, with NEON, there was a synthesis center stood up last summer called Environmental Data Science Innovation and Inclusion Lab. It's a center where they basically are looking at all types of environmental data and training and just doing workshop after workshop and getting PIs familiar with all the resources out there, including NEON. NEON does its own training. So you can't just make it accessible. You've got to do this. We know it. And I think it varies, by facility, by resource, and how we do it. With NEON, for example, there's high-end users that are doing the continental scale research right now, but that's a fairly small community. But there's other people that can do local studies with it, with just a few training examples, right? So you're hitting a nerve that we always pay attention to. It's a huge component of it because access isn't enough usually.
There's another aspect of democratizing. I think you were asking about regional and stuff. So the CXFL, that's pretty specialized, right? The idea is it's taken what's at synchrotrons now and sort of not benchtop, but putting it in a laboratory. And so if they can build this and figure it out and it costs a lot of money, maybe they can teach somebody else once it's done to put it somewhere else. I don't know how many people will want these, but that is sort of the democratizing, is bringing something that was only available in synchrotrons to this. And it may be that pharma companies, in my guess, are going to want to use that, but I'm not quite sure. But that's a different area. It's like access in a different way.
So let me point out one last thing, which is sort of there is a flip side to these, because we have all of the infrastructure programs. And yes, there's been sort of a reasonable response from the biology communities. But for the most part, I think there can be more, right? So in some sense, it's sort of incumbent on the communities outside to recognize that these exist and come to us or to self-organize in some ways and really define what might be needed. So this is sort of what you raised earlier, which is what should we measure and what should we build to measure that? But we can't do that. I mean, and I say this as I'm a former physicist, so I say this all the time. And my particle physics colleagues, they have committees that essentially set priorities for what instrument needs to be built. Yes, those are $5 billion instruments. And that's why they need big pockets, and they sort of have a ranked list of what needs to be measured. But we can do just as well with smaller scale needs, but it's really a question of prioritizing and setting these pathways of design and conceptual design and sort of actually coming up with a final design that can be implemented, which could be anything, right? It could be instruments, could be data sets. So there is sort of an aspect of handing it over to the community to really self-organize and come up with ideas, right? And the bioeconomy can be one focus area, but I can think of probably, and you can think of several, but it's really important to sort of recognize that these avenues are there that can be exploited.
All right, that's been a really good conversation. Yeah, I mean, it's interesting what you said about another thing when he talks about self-organizing. I remember that this used to be a great way that people would write their PUI grants for their broader impacts. They would partner with people and go for the summer and work and learn how to use something and bring it back to their home institution. So that's the kind of supplement that could drive biofoundries and use of the instrumentation as well. All right, if nobody else has any questions, thank you very much, Sri. That was great.
Thank you.
Okay. So our last item for today is to prep for our meeting tomorrow with the director and also with Karen Morangel. So Ponch is actually coming tomorrow to meet with us in person. So what we need to do is to spend a little bit of time now thinking about which topics we'd like to focus on. So what we'll do is what we've always done, that we'll have a few people speak on all of our behalves around, I think, given that there's both Karen and Ponch, I think we should probably limit ourselves to 3 topics, not do 4 or 5 as we've done when it's just Karen, because Ponch there's 2 of them to answer the questions essentially, and we've got 30 minutes is our hard stop because their schedules are busy. So I had some thoughts about what we could have as our discussion topics, but I think we need to first settle on the topics and then decide about what kind of points we want to raise, not too many points, and then who's going to deliver these. So what I would throw out there is that the first thing is we have talked a lot about in different settings, both in the breakout and also, I think, following on particularly from Teresa's talk and from what Brent said and so on about the research goals, the R&D aspects of this. And I think we shouldn't lose track of that. We're brought in as a bioadvisory committee, so I would suggest that we kind of go at that as one topic. I know that, for example, in our breakout group, I thought it was interesting, all the breakout groups went in different directions. We tended to keep on coming back to what are the basic research questions in a broad sense. So I'll just tell you what. So I think that should be one of our topics. I think we've come back again and again to the workforce, accessibility, the biofoundries. You know, these are topics we brought up with Karen last time. She was very responsive to. Suzanne Barbour has brought this up before. Maria Oriati's brought this up. Kennedy's brought this up, Paul Turner's brought this up. I think it's very, very important in terms of it's not just an inclusive workforce, it's a complete workforce to drive the bioeconomy. If we just keep doing what we're doing, we won't have the workforce for this. I think that's kind of obvious. I would suggest that we don't dig deep into the current cost of graduate education crisis. This is being handled separately by the NSB. Thank you. So we don't want to waste the small amount of time we have on something that Karen, Ponch, and other NSF people in this room are already dealing with. I think it would be wrong of us to not mention it, but we don't need to highlight it. I think everyone knows that there's a challenge here, and I think we can simply state that it kind of the challenge is to find additional innovative ways to train people, whether that be by, as Tom said, stackable certificates, whether people have mentioned the European model where you can do essentially a research master's as an extra year straight on after your bachelor's degree. Someone talks about the shorter PhD. That's the UK model. I didn't take a single class. I just did research and graduated. Some other people in this room did the same thing, and yet we're all here still somehow. So there are different, well, Simon's looking doubtful about that training model. I look on his face, but
Did mine in the US.
that's why you're looking doubtful on it. So I think that we should come on that in the workforce. And now with the third topic, tomorrow we do have a breakout group on partnerships, and partnerships has come up, and there are a lot of different ways to look at partnerships. And
Workforce, it's workforce.
breakout group 2, well, it talks about partnerships to achieve NSF strategic goal 2.
Yeah, I think you're looking. Sorry, did I make an?
But I just pulled out this point because I think that we talked about partnerships. Okay. Last minute edits. It's all workforce.
Follow after.
So we're not going to talk. There's not going to be anything. She's not going to talk about partnerships.
She's going to talk about the report that was written.
Okay, so we have some workforce topics, and whoever leads that will get even more to discuss tomorrow. But I think we've got enough to talk about, to think about, that will help to clarify. So what do you think? I'd be interested to know what people think about those first 2 points and what people think a third point could be that we might discuss with the leadership of NSF when we have them for 30 minutes, given the focus of this meeting. Paul.
Well, maybe I just want to go back to the first topic you suggest.
Yeah.
And I'm trying to articulate it in my head and align it with what we had been talking about. And that might be aligning our basic research goals with this important emphasis on growing the bioeconomy.
Yeah.
And basically we need to understand and elucidate how the world works now as
Yes.
essential for developing these solutions in a changing world. You know, as the world changes on us, climate change. And addressing things where the bioeconomy.
I had your name written down, Paul, and I really hope you're not going to tell me you're not available tomorrow.
No, I'm here.
All right. So I would really I thought you were really eloquent about that, Paul. I mean, Maria got passionate about
so
one
was
thing.
Maria, though. I mean,
so
many
was
of us in
Maria,
Africa
but
went,
we were all,
Maria-
it was a very passionate group. But the reason just to give some context to everybody, Paul, I think you're someone who's you do a lot of the first part of that, but you are also in touch with the biotech side of it. Yeah. I don't know if Maria is or not. I'm not. I don't think Barbara is. So that's why I thought you would be the right person for this, because you you
I'm happy
framed
to.
it very, very well, I thought.
Yeah, and I've talked with Ponch about this, so like when he
All right.
visited our institution. So I'm okay with that thing.
All right, perfect. So that will be the first topic, which will be, so what did you call it, Paul? I think you had a good, it would be
I'd said something like aligning our basic research goals with this important emphasis on growing the bioeconomy.
Yep.
And elucidating how the world works now. I think I was not the one who said that. That was either Uriarty or somebody else who, yeah, we got to figure out how things actually work in nature. As we're trying to.
I'll send you all the notes that I'm getting from people from that.
Thanks.
So that's great. I think it's good. I know that Susan wanted to make sure that we brought up the basic research component still as part of this, of what we're talking about.
You guys to do what you want to do.
Yep. Well, I'm using you as a surrogate for what I want, but anyway, that's what I'm saying. So the second part of this, and I really think we need to come back to this, it keeps on coming up in different contexts, is this kind of, what's a very difficult question? I thought that was really good in the last presentation, and the discussion from Shri is cutting edge instrumentation, what it means for cutting edge research, but also democratization and access, and what it means for the larger research endeavor, and what it means for bringing more people into the workforce, and how we talked about this a little bit. We talked about this. I think this is an opportunity to come back into, maybe this is the research infrastructure space that supports what Paul's going to talk about in AIM 1, and then maybe, AIM 1, sorry, I'm writing a grant here. In the first discussion point, and the second discussion point would be around this, and then probably the third discussion point would be around workforce itself.
That'll reflect the order.
Yeah, that'll reflect the order.
15 pages total.
About my broader impact. So a number of people have been pretty eloquent, I'd say, talking about this. I think Kennedy's brought this up in a lot of very valid ways, in a lot of different contexts. I know Suzanne has talked about this before and has brought this forward. So, Kennedy, would you be interested in talking about this, about infrastructure, what it means? I think it reflects, there are different, there are different aspects to this. You know, there's both, I for one was very reassured when I heard the whole story about how that project in Arizona came to be, that it kind of was a organic process that led there. But then, like you said, how does that affect lots of people at other institutions? And maybe sometimes it has to, and say maybe sometimes it doesn't have to. And I'd like, if possible, you could maybe ask Suzanne for some input. You know, because she has input from, what is the committee that she chairs?
CS.
From CS. She has input from CS about accessibility. And we talked about this around bioinformatics and computation, and Tom had talked about AI as well in this space. So these might be different themes that different people could. So if people have thoughts in this space, if you could email Kennedy about this, I think it would be really, really helpful. Are there other topics that people would like to address in that discussion around infrastructure? Haven't got there yet. Well, I got to.
Hey, Mike?
Yes.
Yeah, just maybe just pull a little more on the infrastructure. I was reflecting while listening to the presentation, it was really good, on how transformative the PDB had been, the protein database has been, and how insanely accessible it is to all communities. And I simply wanted to ask a question I didn't until just now. and maybe this is more for Susan or people in the field a little more close. Is there an equivalent for something about the dynamics of proteins as opposed to their statics? Because isn't that the new horizon that should be on the scope of NSF, and that make that a sort of a dynamic protein database?
Yeah, I think that's where research today is, but I don't think it's at a point where a database. There are some people in UW, Valerie Daggett, right, who have their dynamomics or whatever.
Yeah.
Yeah. So, but I think it's unlike structure, whether it be NMR or crystallography, where there is sort of ground truth. And so the PDB was like, okay, we have structures, we can quantify their reliability. You know, Helen Berman said, let's put this into this database. I think we're still at the research of what is the accessible conformations of a protein, which is the question. But if I could add about the PDB, I think it's an amazing accomplishment. And if it would never have happened without something like the NSF to support it. That's never going to happen at a small scale. But you brought up this issue about AI in the beginning of the meeting, and that what did the PDB lead to? It led to AlphaFold and Rosetta Fold. And actually, the original AlphaFold was not available to everyone, right? Google DeepMind did that. And in response to that, people like David Baker said, we have to make, once it was done, it was apparently, I don't understand it, but not so hard to replicate. And so David Baker did Rosetta Fold because everybody needed to have access to it. And that also brings up what you brought up earlier about AI and having access to the availability. But I do think to stress that the PDB is an amazing success, in my opinion. So thank you for bringing it up.
Yeah, and Paul, yeah, I'm elaborating further. It's not just that it was amazingly successful and absolutely needed in ground truth. It was accessible. And I think that was that's the special part about it. And maybe the future is more on dynamics with some ground truthing. Yeah. I think that's good for NSF to think ahead of the so skate front and see where it goes.
Thank you. One other thing about the PDB was the enforcing. Everybody had to put that in, and without the funding agencies saying you're not going to get funding unless you make it accessible, it would have never worked. So it is a real paradigm that we should look to, and we often don't take pride in things that were done 20 years ago, but we should.
Yeah, I was gonna. It's good you brought that up, Tom, because I was thinking about the PDB. It's like something we all take for granted in a way that. The other thing it's interesting to me related to PDB was the point when structural genomics was happening, where you could just send a request, and you might get your structure back. That was really quite something in that phase of structural genomics. That was an infrastructure implementation that everybody could suddenly use. I mean, I don't know if anyone else did it. I remember I got the email like the structure's available. It's like, what is this, Christmas? I can believe it. I mean, I just sent them an email with a record number of a gene. So
just add, and that somebody asked earlier about who knows about the instrumentation, and that you would even know that it would be something you might use. Like, how do you get that out there? And I think that, again, one could look to the PDB. Who cared about structures in 25 years ago? Structural biologists, right? Nobody had this view that people would have gene sequences and be able to say, oh, what does it look like? Why do we care? So that sort of is another paradigm about how only the nerds within the field really care about something, but then you realize it bridges. And I don't think there's anyone that doesn't, any biologist or chemist that doesn't look at what the three-dimensional structure might be of their
I think that's
product.
It's kind of why I asked Kennedy if he'd present, because I thought Kennedy's point was really, really well made about like, that's great. Who can get to it? Who can use it? And PDB is a good answer to that.
Right. And who knows that it might be something.
Who knows that it's there, yeah. sorry. So for the third point, we're gonna cover workforce, and Karen is gonna ask Suzanne if she will lead on that. As you all know, Suzanne has been very eloquent about this at previous BioACs, and the points she raised are very, very valid. And I hope if you don't all feel the same way, please let me know. But I'd really like her points to be heard again by the directorate about the potential to kind of be even more inclusive in how the workforce are engaged across different schools, across the system. And because she's on two different, on us, on BioAC and on another group looking more specifically at this, I think she's exceptionally well placed to represent us, unless someone else feels strongly they'd rather speak. Okay, so one last question. Are there any other topics which fall outside these three, which you would like me as chair to raise with the director tomorrow? Remember, we're being recorded when you respond. Yes, Gretchen.
Well, there was just one point that kept coming up in breakout room 3, and that was extreme urgency. So I think that the extreme urgency in coming up with solutions to issues of climate change, and somehow it's so urgent that the tie-ins with commercial industry and the tie-ins, maybe those, somebody obviously has to pay for it, but it may not be a commercial product that is the result of the research that goes into that. Instead, it may be preventing catastrophe. So I think that I guess I think this is the body that really just has to keep underlining it's urgent. We have to approach it. Everybody's got to pay and that kind of thing.
So I will lead that as somewhat of a context to some of our conversations for two reasons. One, because Maria expressed exactly the same sentiment, and in breakout room 1, what we talked about was that yes, there is extreme urgency, but essentially the bioeconomy doesn't happen if you don't look at this. It's all kind of a moot point. So, and this has been a major stated goal of NSF, is to look at, do we call it climate? What is it?
It's called Resilient Planet.
Resilient. So its central theme is the resilient planet. So the bioeconomy doesn't happen in the absence of a resilient planet anyway. So that has to still be there. Tom, did you raise your hand?
Yeah, just very brief add-on to that. I think I completely agree the urgency is there. The urgency is also reflected in sort of a pivotal time where I think the public-private partnerships are really accelerating. I think both philanthropy as well as industry is actually motivated now to an extent that I've not seen previously. And so I just want to add that.
Yeah, that's Karen just said, that's congruent with the TIP directorate. So I'll use that as if everyone's comfortable with that, I use that as a framing for the conversation then.
Can I make a quick comment about that was great that Gretchen brought that up, because I was thinking of, maybe it was Maria or the other Maria, one of the Marias said, the resilience of our coastal systems, with the loss of some species along coastal systems in the Atlantic seaboard is certainly that protects us against environmental disasters. So, in terms of ecosystem services, I sort of lump that in with, we need to understand those systems and keep them in place. I don't know, there's some way to tie that in with bioeconomy that came into our discussion in our breakout group. So I was just going to try and sneak that in with some of my
I won't steal that. Don't worry, Paul. If you I won't mention.
Oh, no,
it.
no, sorry. If you were saying you want to say it, then all.
No. I'm not going to be very, very conscious of time with the 30 minutes. So I really just want to give the 30,000 foot point, and then that's a great segue, I think.
Sure.
You guys should say what you want, but I think we're all, or you are all, circling around the room what we all fundamentally have in our DNA that we need to remember the other scientists in other fields don't, is that we believe that biology provides the solutions to a lot of society's challenges
that's a
right
given.
now.
I mean,
and that's a given to us in that room, in this room. But if you believe that, you need to make sure that we start at that level when we talk. Like that's the given we're starting with. I just want you to remember that's not necessarily everybody's starting point. The realization we all drank the Kool-Aid and know that.
all right. Biology is the answer. I'm writing it down.
I think life is a billion existence proofs.
Okay, is there anything else that anybody? Yes, Maria.
Yeah, so I mean, bringing it back to the point that Paul was going to make, I guess, I don't know if we can be this specific, but I mean, obviously, the way in which the basic research intersects with something like bioeconomy research in the way it's framed, or the TIP directory and so on, in some ways it's a budget issue. So it would be good to hear the director talk about how are those allocations made in terms of putting more money into these partnerships rather than basic research, because I imagine that that's a decision that is explicit. And also from a more kind of procedural issue, it's been fascinating to be in this committee just to hear. I mean, I think biological sciences is so huge, now we keep having this conversation. But what kind of intersections happen between people that work in vastly different areas? And I think there would be a lot to be learned from that. And I wonder whether there's any kind of plan to have a sort of this broader, interdisciplinary group looking at these questions. Because obviously, the technological advances in bioeconomy are great, but where is the person that says, hey, what about, is this going to work once you kind of take it out and try to scale it up or you put it in the real world, right? And I don't know if NSF has given any thought to these procedural issues of breadth.
Brent, Teresa, playing tag.
So I think that part of Casa Bio is to bring people from diverse perspectives. It's not just biologists that will participate in Casa Bio. It will be the engineers. It will be the entrepreneurs. It will be the mathematicians. And everything that we have done so far that we talked about really involved kind of multidisciplinary teams, and they included industry stakeholders as well. So yeah, that's what Casa Bio we hope will do, is we will bring in expertise and voices and from all sectors, both all disciplines and all sectors.
Bio is the home of everything. I like that. It all works there. Anybody else got anything they'd like to bring up? All right. Well, thank you, everyone. Thought that was a really productive day. Thought we got a lot done. And I will see you all. We will see you all again tomorrow, starting at 7 a.m. Pacific, 10 a.m. Eastern. All right. Have a good evening, everyone.